Chapter 9, Section 8, Passive Penetration and Active Transfer in Animal and Plant Tissues. In this section, once more, permeability of cells and tissues will be the principal subject as it was in Section 4. Here, however, the term permeability will be used chiefly in a sense different from that prevailingly used in the earlier discussion. As repeatedly mentioned before, permeability means the capacity of a physiological boundary to allow the transit of solute or solvent molecules from the surroundings into or across a cell or a layer of cells. In many cases, the process can be fully accounted for as a spontaneous diffusion or osmosis, but in other cases, the passage appears to be enforced by energy ordinarily derived from cell metabolism and indispensable for the process. Thus, it has become customary to distinguish between passive penetration and active transfer, both as being aspects of permeability, but which are essentially different in nature, and which have sometimes been designated as physical or physicochemical. Investigation of the Fournier was initiated in intimate association with the beginning of physicochemical interpretation of physiological phenomena. It was the era of Van Hoff, Pfeffer, Arrhenius, and Nernst. The present section will deal mainly with the problems of the physiological permeability, a concept originating in the earlier physiology at a time when one still attempted to describe the action of entire organs in terms of mechanics. So successfully applied to the physiology of circulation, of respiration, and of lymph formation, but then increasingly found to be inadequate in the physiology of urine formation, of intestinal absorption, of glandular action, and of others. In pursuing this line of research, it became obvious that in analyzing the activity of those organs, which accomplish a shift from one place to another of water and of certain dissolved substances in characteristic concentration ratios, one was compelled to differentiate between that part of the functional result which might be due to passive penetration and that part which might be due to active transfer. This problem has been solved, as yet in only a few cases, partially owing to the simple fact that sometimes it is difficult to define what is active and what is passive. Active transfer usually is manifested by the establishment of an unbalance in concentrations. Solute or solvent molecules are shifted uphill aga into diffusion gradient. They are accumulated as the result of osmotic work, enabled by the liberation of metabolic energy. However, concentration gradients may also arise without metabolic intervention, for example, when a Donin equilibrium is established. Consequently, the simplest way to distinguish between active transfer and passive penetration seems to be to cut off the source of energy by a metabolic poison, by anoxia. Procedure can fail to give a clear-cut result. Another criterion is based upon the fact that the Donin distribution applies to ions but not to non-electrolytes. Let us consider some examples. 1. The classical example is the distribution of ions arising when a solution I of a colored electrolyte NaR is separated from a solution O of nickel by a membrane which is permeable to RaNCl. Impermeable. The resulting equilibrium can be expressed by the equation Naginao equals chloi I and O, indicating the inside and the outside solution. Gl cli now, pages 68 and 79. The corresponding inequality of cation and anion distribution has been ascertained in a great number of physiological systems, where two body fluids were separated by a physiological membrane, for example. The fluid content of the red blood corpuscles and the scrum separated by the plasma membrane, or serum, and edema fluid separated by the capillary wall. This latter system has been analyzed, among others, by Hastings, Salveson, Sendroy, and Van Slyke, with the two. A somewhat different form of Donin distribution is illustrated by the following experiment. See also 254. A dried collodium membrane separates two solutions, one containing M1400K2, so 4 plus M10H2, so forth, the other M1400 K2S4. The two solutions are kept in osmotic balance by addition to O of non-penetrating glucose. The membrane is permeable to K and H, impermeable to so forth. In other words, at the beginning of the experiment, K and Co are equal. I about 10 times greater than L, Since H and O are reciprocal to each other, the final ionic distribution should be: after 14 days of ionic exchange across the membrane, the following concentration ratios were found. In other words, K was shifted from outside to inside against an increasing concentration gradient, whereas in the opposite direction, it fell to a value below the initial value. Thus, the two ratios, which at the beginning were highly different, approached each other toward the same theoretical value. This experiment has suggested interpretation of the conditions in muscle fibers as a Donnan distribution, because Ki in muscle is about 30 to 40 introduction. The problem's times greater than HER and high greater than as a matter of fact. This concept is substantiated by the fact that the fibers have appeared to be selectively permeable to K and to H, impermeable or at least much less permeable to anions and to Ni, and that by changing and O respectively, the ratio Kirchhoff can be displaced in one or the other direction. However, the quantitative agreement between theory and experiment is not very satisfactory. As might have been anticipated on account of a number of complicating circumstances caused mainly by metabolic reactions, percent which are released by unphysiological concentrations of H and K, 309 and 33. Another variety of Donnan distribution due to a selective ion layer of carbon tetrachloride is placed between a solution of 0.2 norm sodium benzoate, I, which is kept at a fairly neutral reaction by the agitation of buffer, and a solution of 0.1 norm. NaCl, the membrane is impermeable to Na, and behaves as though permeable to H and benzoate, since the undissociated free benzoic acid is soluble in the organic solvent. After 2 hours, POO was found to be 3.8, Phi 6.9. In other WEXRDs, the concentration of H outside has increased several hundred times, whereas that of benzoate has fallen with the concentration gradient to a lower value. This model was investigated by TRL with the idea of correlating the process, governing the accumulation of H ions to that involved in the production of gastric juice. Although this model was found useless, since the gastric glands are able to raise the H concentration about 3 million times above the blood level, P599. Attention may be drawn to the fact that the formation of the slightly alkaline secretion of the pancreas has been attributed tentatively to Donnan distribution across the epithelial cells. It follows that with regard to physiology, the cases of Donnan equilibria mentioned under 1, 2, and 3 can be identified as such, at least in principle first by the distribution of the penetrating ions, the ratios of the various cations being identical and being reciprocal to those of the anions, and second, by the failure to show a relationship, the establishment of the unequal distributions and metabolic reactions. In other words, the Donnan distribution of ions belongs to the category of passive penetration. Finally, another form of passive penetration should be described, which likewise leads to an unequal distribution, but although dependent upon a metabolic reaction, is essentially the result of a passive penetration. As mentioned previously, P260, Osterhout has investigated the distribution of the weak acid E28, KLAC was 5.7 between the acid cell sap of Valonia, which by means of the cell metabolism is maintained at H5.8, and the surrounding seawater, of which, by the addition of TACLL or now, was varied between 5 and 10. The result was that, for example, at 5.2, the outside concentration Co of His was found to be approximately the same as the inside. C pound equals 197, whereas, for example, at 850 was about 25 times higher than that inside CoEC 104. The explanation is as follows. At PLA 5.2 outside, H2S is almost entirely present in the form of Uki-associated molecules, which, like those of many weak acids, enter and pass as such by diffusion across the protoplasmic wall, whereas the ions cannot penetrate. At PW 5.2 outside, the concentration of undissociated 2,2S is slightly higher, the concentration of the ion slightly lower than at pH 5.8 diaphanty. However, at pH 850 outside, H2S will be prevailingly ionized, and only the small remainder of molecular H2S can distribute freely until diffusion equilibrium. This interpretation is supported by the fact that, for the entire scale of PLL, the concentration of total H2S inside, unionized plus ionized, was found to be approximately the same as the concentration of undissociated H2S outside, as can be calculated from the concentration of added H2S in its pK. It follows that de facto we are dealing with a passive penetration. However, provided that the natural difference of pH between the cell sap of Valonia and the surroundings is based upon the metabolic activity of the protoplasmatic wall, as it probably is. Each change in the rate of this activity will be followed by a redistribution of the weak electrolyte in one or the other direction, and will produce the A corresponding behavior can be demonstrated with the salts of weak bases, for example, of alkaloids and of basic dyestuffs. For instance, neutral red delivered to the outside of the proximal tubules of the frog kidney in a neutral saline solution by way of the renal portal vein, while an acid fluid flows from the glomeruli, appears highly accumulated in the tubular lumina, thus displaying the picture of an active transfer. In contrast, an alkaline fluid passing from the glomeruli along the lumina prevents on the basis of these introductory remarks, an analysis of what in a broader sense has been called absorption and secretion will now be attempted. For this purpose, we shall continue to distinguish between the life processes described under passive penetration and active transfer. In this way, the simpler facts can be made to serve as a foundation for understanding the more complex and less accessible phenomena. In order to facilitate the classification of the findings, the following criteria will BTI set up. A movement against the concentration gradient will in general be considered as indicating active transfer. 1, if the movement is inhibited energy for affecting the uphill shift. 2, if, in contrast to the Donna distribution, cations and anions are transported simultaneously and in approximately equivalent amounts and in the same direction. S, if the shift takes place with non-laurolites. 4, finally, if an unequal distribution of a weak electrolyte appears to be due to a PW difference originating from cellular metabolism. The subsequent analysis of absorption and secretion will be made with reference to 1. Intestinal absorption, 2. Urine formation, 3. Permeability of the body surface, 4. The elaboration of digestive juices, 5. Some remarks about the energetics of the active transfer, the transferring devices and air action, including remarks concerning the action of the chorioidal plexus and the ciliary body. For a long time, intestinal absorption has been the favorite subject for studying absorption. The intestine of higher animals offers for this purpose an extensive layer of epithelium, covering many folds, each of them often providing space for a great number of villi. This epithelial membrane bounds the wide cylindrical cavity of the lumen, which in an experiment can be easily filled with a solution, the absorption of which is to be investigated. This procedure would approximate the normal conditions, since in the higher animal, absorption deals with solute plus solvent. The delicate epithelial layer, about 25 micra in thickness, is interjosed between the solution and the subepithelial tissue, which to a large part can be considered fluid, since it includes the blood plasma in the capillaries and the lymph in the interstitial spaces. This subepithelial fluid, though small in volume, can be thought of as representing a much larger volume because of telic continuous circulation, like sweeps away substances, entering this space fluid fairly constant. Local variations of concentration in the experimental solution, when it comes into immediate contact with the epithelial membrane, in filling the narrow spaces between the folds and between the villi, are diminished by the stirring effect of the movable villi. These complicating conditions raise the question whether absorption experiments are capable of permitting a definite decision concerning the relative importance of diffusion and osmosis, or whether superimposed processes may cloud the interpretation. Regarding the methods proposed for studying intestinal absorption, The nearest approach to a scheme suitable for fruitful physiochemical analysis seems to be the loop experiment, although more than one obvious objection can be made even to this. In conducting such an experiment, the body cavity is opened and a section of the intestine is isolated by ligatures, rinsed with wringer, and filled with a measured amount of the experimental solution. The conditions resemble those in a diffusion experiment, in that one can follow by chemical methods the passage of substances across the constant wall area of the loop during a definite length of time at the fairly constant temperature of the circulating blood. If one compares isosmotic solutions introduced successively at the beginning of each experimental period, the concentration gradients across the separating wall are identical. Since the concentrations beyond the membranes are near zero, or rather constant, providing the solute in question is not one of the normal constituents of the body fluids and possibly subject to metabolic concentration changes. Absorption under the above conditions can be expected to obey Pick's law of diffusion, see Sec 1, Chap 1, and to show the following characteristics. 1. The rate of absorption, that is, the amount of solute leaving the loop contents in a certain time, is in a linear relation to the concentration. In other words, the percentage of absorption is constant. When several substances are compared under identical conditions, individual differences in the rates of absorption should appear to correspond to Pick's diffusion coefficients. However, it should be anticipated that the method described does not permit an exact confirmation of Pick's law to be obtained. Impairment of the vitality of the delicate epithelium. Through mechanical insults following the repeated filling and emptying of the loop, uncontrollable changes of the blood circulation, the effect of the narcotics, and other factors are unavoidable causes of irregularity. For many special problems, it would be very useful to change the loop method in such a way as to provide the isolated loop with its own artificial circulation of an adequate perfusion fluid. This procedure has been tried by O'Neill, while additional methods have been proposed, which avoid some of the faults of the loop method but introduce others. The procedure of Corey has often been the method of choice in comparing the absorption of several substances, or absorption under other varying conditions. Groups of rats, which by their anatomical and their physiological behavior resemble each other as much as possible, are fasted, then fed the solutions by stomach tube, and killed after a suitable time. The total gastrointestinal tract is excised, and the unabsorbed contents analyzed, percent abs percent. In the following, some experimental results are listed in an effort to describe the absorption of non-electrolytes, of electrolytes, and of colloids in terms of physical chemistry. There are not many observations concerning non-electrolytes, although they are generally foreign substances and, as such, especially suitable for experiments because 1. Their concentration in the body fluids outside the epithelial layer is 0, and 2. Metabolic reactions supposedly do not interfere with the course of diffusion. Table 41 refers to experiments on rats, where the influence of concentration on the rate of absorption was studied. Obviously, the course of these experiments, in at least a rough approximation, follows the predictions of Pick's law, even though, irrespective of the various aforementioned interfering factors, the regular diffusion probably was partially obscured by osmosis across the intestinal wall, due to the varying osmotic unbalance between the experimental solutions and the body fluid. Another group of experiments deals with the rate of absorption of different non-electrolytes presented in equimolar concentration. Comparing such experiments to the corresponding observations with artificial membranes, the results provide a clue to the physiological diffusion coefficients. Table 42 gives an example of the absorption of various aliphatic acid amides. Percent ab is as explained in Sec 1, Chap 1. In agreement with Einstein's law, the diffusion coefficients can be correlated with the molecular volume, MV, or they are the same whether determined in free diffusion or measured by diffusion through the pores of a sieve-like membrane, provided the pore diameter exceeds markedly the size of the molecules in question. The table thus shows that on the whole, the observations with the acid amides meet the theoretical requirement. However, the differences of absorption rates observed are far greater than the differences in the rates of free diffusion or of diffusion through membranes with wide pores. Diffusion coefficient ratios, as great as 1, are considered very large in free diffusion. For instance, typical values obtained for mannitol MV189.2 equals 0.5 for colander. This can be interpreted as being due to the fact that in sievedic membranes, for example, in the dried collodion membranes, the pores fail to have a uniform diameter, but rather show a variety of sizes. It follows that for larger molecules, only a small number of pores may be available, that is, a small portion of the total pore area. While the smaller molecules penetrate it, is obvious that below a certain minimum pore diameter, a sieve membrane will appear to be impervious to certain substances, which among others may be present in an experimental solution. In this case, they will prevent the absorption of the solution to a degree depending upon their partial osmotic pressure by inducing an osmotic current to flow into the lumen of the intestine. To a smaller extent, medium-sized molecules will produce the same result. This effect is the chief factor in the purgative action of a copious administration of such water-soluble substances as disaccharide sucrose, lactose, in contrast to monosaccharides. The osmotic increase in volume is accompanied by a distension of the intestinal wall, which initiates a peristaltic wave, causing expulsion of the liquid contents. But there are many observations regarding the absorption rate of non-electrolytes in equimolar solutions, which by Noam and show a correlation to the molecular volume. 2 examples are given in the following table, referring again to the absorption of aliphatic acid amides percent abs distributylic oil. It is evident from this table that 2 substances of nearly equal molecular size, succinimide and malonamide, can differ considerably in their absorption rate, and even that, as in the case of lactamide and valoramide. Larger molecules can penetrate the absorbing membrane with greater speed than smaller. The cause apparently is relative lipoid solubility, which with 2 of the experimental substances, lactamide and malonamide, is extremely low, and with the other 2, succinimide and valoramide, although still very small compared to figure 23 to 32, is about 50 times greater. It may be concluded that the factor of lipoid solubility plays the same role in tissue permeability as in cell permeability, discussed in extenso in On as yet. However, pharmacology records a great number of separate euertinent observations, such as the rapid action of narcotics, many alkaloids, hydrochloric acid, and others following oral administration. Another factor besides molecular volume and lipoid solubility in determining the rate of non-electrolyte absorption seems to be of a more chemical nature. On several occasions, it has been mentioned that the concept of light or insolubility is rather vague. But just this vagueness is of great interest and will possibly become more and more so, if each slight shade of lipoid solubility can be correlated with certain chemical or physicochemical variations, and can be shown to reflect certain structural or functional manifestations of living entities. Already Overton, when he developed his theory, has stressed that cellular lipoid is not fat, but fat-like. Since it appears to be a mixture of a group of substances, each individual component of which is capable of conveying to the mixture, according to its relative amount, special characteristics, either as a solvent or as a substrate of some kind of chemical reaction. See pages 234. Today, there is reason to believe that the same cells, or the same physiological unit, may harbor a number of lipoids, each of them involved in a certain function of life. For instance, it was mentioned earlier in discussing the lipoid theory of narcosis that the depression of excitability by a narcotic seems to be due to the presence of a substance resembling oline alcohol, P358. Another example of the significance of small variation in lipoid solubility appears in some further experiments concerning the intestinal absorption of non-electrolytes that are only worth mentioning here because they throw light on the significance of such light differences. Referring to the findings shown in Table 43, the conclusion can be drawn that substances which are practically lipoid insoluble, as well as of nearly the same molecular volume, can differ enough in lipoid solubility to cause definite differences in the rate of absorption. As a matter of fact, Jacob's species, Wilbrant, Ildflar, and others with many kinds of plant cells have observed a contrasting behavior regarding the penetration of aliphatic acid amides on the one hand, and of polyhydric alcohols on the other. Under the same conditions, one group of erythrocytes or of plant cells is found to be more permeable to the acid 0K, HD flare, or DCVAT. GIZS 6053 1932, 62 355. Possibly this can be accounted for as being due to differences in the individual cell lipoids, Höber, as a matter of fact. Collender and Barlund have corroborated this idea by model experiments showing that, for instance, the preferential penetration of a number of acid amides into cells of Cara compared to Rho is imitated by the greater relative dissolving power of olive oil plus oleic acid in contrast to pure olive oil. This is possibly due to the amide radical, although the basic character of the aliphatic acid amides in aqueous solution, as well as in organic solvent solution, is very weak. The reverse physiological effect can be expected to be produced by adding to the olive oil an organic substance, which increases the basic affinities of returning to intestinal absorption. The acid amides were found to pass the absorbing membrane markedly more slowly than the polyhydric. So far, absorption has appeared to be like a passive penetration. We now turn to the absorption of salts. On reviewing the history of studies upon intestinal absorption, one is immediately struck by the fact that during the more than 40 years succeeding the enlightening research of our Heidenhain about absorption, secretion, and lymph formation, and his endeavor to differentiate between physical and physiological factors involved in these properties, the major part of the work in the field of absorption was devoted to the study of salt absorption. This historically understandable fact has hampered progress since the time being immature for the useful application of physicochemical concepts. Great complications inherent in an analysis of the penetration of a membrane by electrolytes could not be perceived. These complications grow out of the following facts. 1. In the case of diffusion of a salt, we are dealing with diffusion of at least two substances, the anions and the cations, the mobilities of which influence each other through their electric charges. See Sec 1, Chap 1, 2. In solutions of weak electrolytes, for instance, the salts of fatty acids, see later, hydrolysis takes place which may affect membrane diffusion, either by shifting the pH in one or the other direction, and thus altering the membrane structure, or by the fact that the undissociated molecules of the free acids or the free bases are able to pass through the substance of the membrane, which is impervious to the ions, a colloid cation or a colloid anion, present on one side of the membrane and too large for penetration, absolute impermeability, will cause a Donan distribution, CP 79, for as has been shown, especially by TRL. Some sort of Donan distribution, diffusion effect, also will appear if two salt solutions are separated from each other by a sieve membrane, the pores of which are wide enough to allow the passage of each of the ions, but the passage of one of which is retarded by a relatively low mobility, relative impermeability. 5. The picture of salt absorption frequently is much obscured by the entrance of salt from the tissues. This is due first to diffusion of salt into the lumen from the body fluids, particularly from the blood plasma circulating through the capillaries of the mucosa. Second, to the secretory action of the intestinal glands, the product of which contains salts, especially natal. The existence of the first process has often been denied, for the reason that there is evidence that various normal absorbing or secreting membranes exhibit only unilateral permeability, even to substances as highly diffusable as are, in general, the inorganic salts of the eulasma. As a matter of fact, it will be shown later, p. 542, that in the intestinal W all forces arc, evidently at work shifting particularly the highly diffusible salts from the lumen to the body fluids. On the other hand, there is also reason to believe that the entrance of salts into the intestinal fluid is, partly at least, attributable to diffusion. For instance, Goldschmidt and Daytonhave introduced into the colon of a dog NaCl solutions of increasing strengths 0.04 to 0.9%. The amount of NaCl J-acing into these solutions from the surroundings was found to be smaller, the higher the concentration, and to become zero at a certain concentration level, about 0.6% NaCl. A equals 0.13040. Above this level, U passes into the blood. Most obvious is the bilateral movement of ions in recent experiments of Vischer and associates, showing that radioactive sodium ion present in the intestine of dogs in a mixture of isotonic NaCl and isotonic NGS, 4-CP. 542 is shifted against the NR gradient from the intestine to the blood, but that Na, after intravenous injection, simultaneously passes along the concentration gradient into the intestinal fluid. Accordingly, Pendleton and West DUE have found that after the intestine has been filled with normal saline, urea appears in the solution and rapidly rises to the blood level. Furthermore, after an intravenous injection of urea, the intestinal fluid again comes to a concentration equilibrium with the blood urea, and as the urea percentage declines in the blood, the urea level in the intestine falls off correspondingly. 6. Finally, in trying to unravel the multitude of physicochemical factors hampering the effort to clarify the distinction between passive penetration and active transfer, osmosis must be mentioned. Although it is by no means specific to salt absorption, but plays a role in each kind of exchange across the absorbing membranes. The significance of the association of osmosis with salt movement is obvious. For example, in the following experiments of McDougall and Verzara regarding absorption by the rat's intestine in nearly millimolar solutions of glucose and xylose, both strongly hypersonic to the rat's blood, about 0.3 molar tables sheet. Osmotic pressure equal to 3 cc of nearly equimolar hypertonic solutions were introduced into the intestinal loops. After 1 hour, they had become approximately isotonic with the blood, the osmotic pressure of the remainder in the loop being expressed in percent NaCl. This was evidently due partly to an influx of water, partly to the entrance of NaILL. Glucose appears to be considerably more rapidly absorbed than xylose. This difference between the two sugars will be discussed later, P544. It explains the greater increase of volume, as well as the greater amount of NaCl entering the xylose solution, both contributing to establish osmotic equilibrium with the blood. This final isotonicity between blood and intestinal fluid has been observed very frequently and regularly, disregarding whether the experiment is started with a hypertonic and isotonic, or a hypotonic solution, including this discussion of factors to be considered in order to account fully for the great variety of findings in absorption experiments, including such factors as, so far, have not received much attention, Sec 2 to 4. We turn to experiments approaching the problem of passive diffusion versus active transfer, by way of a procedure similar to that which appeared to be useful in studying the absorption of non-electrolytes. This procedure was distinguished by the avoidance of great osmotic gradients and the use of short absorption times, comparing the absorption of anions administered in isotonic solutions as sodium salts. The absorption rates follow roughly the order of Cl, Bri formulate, atat propionate, deuterate, valerate, capronate, CeNO3, lactate, heptote, 7, caprolate, Cg, sulfate, phosphate, ferrocinide, tartrate, citrate, fluoride. This series looks, at least with respect to the inorganic anions, as though diffusion were the Juke-retinionate factor, since the ionic mobilities decrease in a corresponding succession. This interpretation also contributes to the understanding of the purgative action of sulfates and phosphates and other enzymers of this group, which, like the disaccharides, in higher concentration and because of their low diffusibility, produce a large osmotic influx of water into the lumen, CP534. But there are several other factors involved in determining the absorption rate of the salts. First, they may damage fly tissues. thus oxalate. And still more fluoride often are visibly injurious to the intestinal mucosa and produce an irreversible functional disturbance, probably caused by a commonly precipitation of Ca, which is accompanied by a loss of adhesion between the epithelial cells so that this layer is readily detached and disintegrated, CP305. Also, the mildit reversible purgative action. Which is observed not only with sulfates and phosphates, but also with malate, tartrate, and citrate, has been referred by Wallace and Hishney to the relatively low solubility of their K-salts or to the deionization of I by these anions. Indeed, some lack of TA is not unlikely to contribute to the laxative action by increasing the permeability of the cells themselves to water. Another vit dictue referred to by Wallace and Cushnie is suggested by the striking resemblance of the aforementioned physiological order of anions and the Hofmeister series of anions, PI 93, also 234. This may be thought of as being connected with the Wefferty-1 known reversible swelling and shrinking effect of salts with univalent and bivalent anions upon hydrophilic culture. Owing to hydration and dehydration, see pages 295F, and possibly localized in the intestinal wall at the colloids serving as senqueating material in the interepithelial pathways of the ions, CP540. Finally, it is known, mainly from the studies of Donnan and Potts and of McBain, that in the series of the alkali salts of fatty acids, starting with acetate, the surface tension of their solutions rather abruptly falls off beyond valerate or caprinate, so that an injurious cytolizing effect can be expected to start with heptote and caprylate, as is in reality indicated in the detrimental influence on absorption. See further. With regard to the absorption of cations, there is not much difference in the absorption rate of the chlorides of NiK. NH4Ca markedly slower is the absorption of MgCu and MgClO, both well known as purgatives. Their diffusion rate is low, due partially to the low mobility of the Mg ion, partially to incomplete dissociation and formation of molecular complexes. Concerning the absorption of univalent chemically indifferent neutral salts, as influenced by the presence of certain other solutes, see pages 540-12, AA or CRAT. Intercellular or transcellular absorption. This problem first became evident when lipoid-soluble substances were found to be absorbed by the intestine at a higher rate than lipoid-insoluble ones, even though their diffusion rate was lower. This presumably indicated a direct passage across the lipoid components of the cell body. In order to solve the problem, dyestuffs were chosen, which as basic dyestuffs, usually the hydrochlorides of diester bases, in general are vital stains. They enter the Lifting cell because the Y are lipoid soluble in contrast to acid dyestuffs, mostly sulfonic acid dyestuffs, which in general are indifferent toward the cells. Chap, EF dogs and frogs, best as tadpoles, are fed the dyestuff. In the intestinal epithelium of the tadpoles, the basic dyes are found either within intensely stained granules or distributed more homogeneously throughout the protoplasm, or both. Acid dyes in general fail to be visible in the cell body as well as in the intercellular cement, but are proved to pass the intestinal wall since they appear in the urine, although often slowly and scantily. Is it permissible to conclude that the entire group of lipoid insoluble substances, non-electrolyte as well as electrolyte, do not pass the epithelial walls, but rather the minute specks between the cells do not even foodstuffs like sugars? Amino acids, inorganic salts, area of the cell body. These questions have been approached in experiments with dyestuffs in the following way. Fresh excised epithelial membrane of tadpoles vitally stained, for example, by toluene blue, was placed in a solution of ammonium molybdate, which precipitates basic dyestuffs. The effect was that the color faded in the granules and escaped from the cell to its surface, and formed a dark blue envelope around the cell body. This indicates that, like many other salts, molybdate cannot enter the cell, but sucks the dye into the intercellular space. This microscopic picture fails to appear if mercuric chloride or picric acid is substituted for the molybdate. The reason is that, in contrast to molybdate, these two compounds are lipoid soluble and are precipitants of the dye, so that the dye can be fixed in loco inside the granules. Furthermore, after the vitally stained epithelium has been killed by osmic acid or by formaldehyde, molybdate can enter and preserve the original picture with the dye stuff stored inside the granule. Now, it certainly should be decided whether the behavior of molybdate can be generalized and accepted as a proof that the absorption of the lipoid insoluble substances is restricted to the small intercellular spaces. In this respect, it is rather well established that some lipoid insoluble foodstuffs, that is, certain alkali salts and some sugars, pass across the intestinal membrane not by diffusion but by active transport, which is associated with cell metabolism. This will be shown more convincingly to take place with certain single cells which, sufficiently supplied with energy, are able to shift inorganic salts selectively and against a concentration gradient, see Chap 36 and 38. Thus, we leave so far unanswered the question of the importance of intercellular absorption B. We will return to it later. Only one point must be added, which has been neglected as yet. There is a group of lipoid insoluble substances which can be assumed to undergo transcellular absorption by mere diffusion because the molecular volume is small enough to allow them to pass the sieve-like cell surface. This may occur with acetamide, ethylene glycol, glycerol, urea, and others, CP233, but has not yet been proved. 6. The part played by colloids in intestinal absorption. Assuming that in many res X, intestinal absorption may resemble diffusion across a membrane, absorption of colloids cannot be expected to occur, except to a very small extent. The apparent absorption of common food colloids can be referred to a pre-aeratory breakdown, mainly affected by the splitting power of enzymes, which even after careful rinsing of the intestine with normal saline can hardly be entirely excluded. On the other hand, there is clear evidence of absorption of genuine proteins in all those cases, where the intake of foreign protein is detected by specific immunological reactions as in food allergy. However, in these absorption processes, where often extremely small amounts are involved in the poisoning effect, the uptake is probably of another nature than hitherto considered, for example, the result of phagocytosis of an unusual leakiness of the mucosa, and of other factors. This nearly total incapacity of the colloidal protein to penetrate the absorbing membrane is of interest in another connection. It has been suggested that the colloido-zerotic pressure of the blood of about 30 millimeters, Hg, due to the plasma proteins, may under appropriate conditions play a part as a driving force for the fluid contained in the intestine of rabbit of Chad, have tested this hypothesis in the following way. They tried to balance the colloid-osmotic pressure of the plasma by filling the intestine with a solution of a non-protein colloid, 6% gum acacia in, 0.9% nacle. According to earlier work, the 6% acacia was expected to be osmotically equivalent to the normal percentage of plasma protein. The experimental results showed no difference between the rates of absorption of the saline with and without acacia. However, this does not disprove the influence of colloid osmotic pressure upon absorption. For according to Dodds and Haynes, NaCl has a profound depressing effect upon the osmotic pressure of the gum, which decreases to only little more than 1/3 of that of the blood colloids. C-third, the problem remains unsettled. Absorption of inorganic salts in presence of slowly diffusing DOS substances. In the introduction to this section dealing with physicochemical aspects of absorption and secretion of dissolved substances, it was I proposed, p. 525, to divide the processes which are coordinated in the physiological functions into spontaneous and enforced movements, first appearing to be mainly diffusion-like. In this special part, dealing with intestinal absorption, we turn now to observations which conflict with the course that could be anticipated for spontaneous penetration. Whether the criteria of active transfer p. 528 apply to their course or not, this has to be tested. After a hypotonic solution of NaCl has been placed in the intestine, its concentration rises until isotonicity has been attained. CP 538. intestinal fluid and blood in experiments with the colon of dogs, Isle, Simmet, and Dayton, have found that if Na2S4 and NACL are introduced simultaneously, the concentration of NACL decreases rapidly, and even can reach the 0 point, whereas the concentration of Na2S4 rises so that the solution approaches the osmotic value of the blood. The absorption of NACL is increased by Burns and Vischer have extended these observations by comparing the behavior of anions other than SO4 when isotonic solutions of the various NAS salts are placed in intestinal loops. Cl enters and its concentration varies during the experiment in a characteristic fashion. As shown in the question arises how to account for this peculiar distribution. Evidently, the chief factor is the difference in the diffusion rate of the ions ingestion, acetate being slower than chloride, and sulfate, phosphate, and citrate still slower. For a number of reasons, see later, it is suggestive to believe that the intestinal epithelium is able to shift EL together with Na uphill against the concentration in the tissue fluids. But this cannot become evident unless a second substance of lower diffusibility is present, for provided nothing but NACL in isotonic concentration were present, and NACL were shifted from inside to outside across the cell layer. Then the now hypotonic solution would become isotonic again by osmosis and so on. A simple COM-E absorption of the salt solution would be the end result without showing any indication of the special driving factor involved. However, for example, with the rather indiffusible, and therefore rather inabsorbable, Na2SF4 present in isotonic solution, in addition to a small amount of NACL, hardly any osmosis would occur, and the NaCl removal would become evident and would continue until the 0 concentration had been reached. This interpretation is in line with older experiments of Katzenellenbogen, which dealt with the absorption of slightly hypertonic solutions containing 0.4% NaCl, in addition to various non-electrolytes. The result was that, for example, in the presence of the almost inabsorbable mannitol, the NaCl constant ration, though from the beginning below the blood level, further decreased, but it increased slightly with erythritol or glycerol due to their smaller molecular volume. A corresponding result has been obtained by Ingram and Vischer, who investigated the absorption of NaCl in the presence of sucrose, and found the salt concentration to fall to 1/3 of the plasma level, although the relative absorption rate of sucrose was only moderately slow. Furthermore, the same authors have continued this work on absorption of mixtures by adding to NaCl chlorides with polyvalent cations. The likelihood of an effect analogous to that with polyvalent anions is not great, since the diffusibility as determined from the ionic mobilities is not very low, and since, in addition, many polyvalent cations are poisonous. Indeed, the results gathered so far with the bivalent NGKMN and the trivalent CoNH36 prove the EA to fall below the plasma level, but not so much as Kiel in the corresponding experiment with polyvalent anions. According to these 3 sets of experiments, under special and understandable circumstances, that is, in presence of substances with low diffusibility, NaCl or better NaL and Tl, can be shown to move against a considerable concentration gradient from the intestinal lumen into the surroundings. For an explanation of this phenomenon, 2 viewpoints may be considered. First, for several obvious reasons, it seems suggestive to refer for interpretation to the Donnan distribution or to TRL's diffusion effect, see Sec 1 Chap 1. However, Jane provided that a more thorough study of the influence of non-electrolytes confirms the existing results. It will be impossible to explain them on the basis of the purely ionic V2. The conclusion might be drawn that an active transfer was demonstrated by the special experimental conditions. This primarily postulates the liberation of energy, which is linked with the shift of ions, see P520. Following this line of thought, Ingram and Vischer have studied the influence of metabolic poisons like cyanide, hydrogen sulfide, fluoride, or arsenic upon characteristic changes of concentration of TLF. So for an Na in the intestinal fluid, one typical result is reproduced in figures 58 and 59. There it appears that in the presence of the poison, the impermeability to sulfate disappears, that the normal rapid fall of Cl to zero is reversed to a rise toward the plasma level of 100 mmol, and that the sodium concentration, which at the beginning is as high as 200 mmol, likewise approaches the plasma level. Thus, the picture has been completely changed. However, the results permit no more than the conclusion that evidently the poison has abolished the normal cellular limitation of ion permeability, so that unrestricted diffusion can occur. This is not then a direct demonstration of active transfer in this complex system. However, it will be shown in the following chapters that there are systems in which the relative rates of diffusion of concomitant substances do not play such a decisive role in identifying the active transfer, and in which the correlation between this transfer and the liberation of metabolic energy is unambiguous. It may be that substances will be discovered which are specific inhibitors of the energy-providing reactions, for instance, more like fluorescein in its specificity than like iodoacetate in its general impairment of cell structure, CP. 548. 3. The absorption of sugars. Sugars are lipoid-insoluble non-electrolytes. It can be inferred, therefore, that in their rate of absorption, the molecular volume will be a determining factor. However, the fact that we meet here for the first time physiologically important organic substances may suggest a special behavior. As a matter of fact, more than 40 years ago, it was pointed out that, on the one hand, the lower absorption rate of disaccharides compared to monosaccharides can be correlated with the greater molecular volume and the lower diffusion rate, respectively. But on the other hand, glucose is absorbed at a higher rate than other figures 58 and 59. Influence of poison upon the distribution of Cl, so for, and nay, between intestinal fluid and blood. Hexes I'm studying then a series of monosaccharides with various methods, ankle different animals. It has been found that, apparently incompatible with the diffusion law, not only hexases fail to be absorbed with equal speed, but pentases pass even more slowly than the hexases. With different methods, Nagano, working on dogs, Corey, Schudy, and Wilbron and Lost on rats, have obtained the following relative values: galactose 100, fructose 44, mannose 33, sorbose 30, isolose 30, arabinose 29. These observations have been supplemented, and more light has been thrown on the physiological nature of these differences by the studies of MacLeod. McGee, at using the excised loops of cats and rabbits, submerged in normal saline and filled with solutions of sugars, the penetration of which was measured by analyzing the outside solution. This simple procedure has yielded the following results. From surviving loops of equal size, glucose passes faster than xylose, but after the mucosa has been injured by heat or by fluoride, the contrary is true. Even lowering the temperature to 0 degrees has the effect that xylose leaves the inside fluid slightly faster than glucose, but the normal ratio can be reestablished by warming the loop to 40 degrees. And further, the temperature coefficient of penetration of glucose between 0 degrees and 20 degrees is about the same, irrespective of whether the intestine is alive or dead, whereas between 20 degrees and 40 degrees. This coefficient is much greater in the living, CP31 thread. All these facts point to the conclusion that the absorption of xylose is a non-selective physical process, whereas in that of glucose, the peculiar properties of the living structures of the intestinal wall are involved. Another contribution to this interpretation is the observation of Corey and Goltz, that the different sugars are absorbed at equal rates from the peritoneal cavity, the walls of which, in this regard as well as in others, behave like an inert physical membrane. Another remarkable result of the experiments of Occhinachi, MacLeod, and McGee with excised loops is the following. 2 loops, one surviving, the other killed, are filled with glucose saline and placed in saline, and the rates of penetration are measured. From the fluid in the dead loop, there appears more glucose outside than from the living. This is in agreement with the fact that the germinability of all living cells increases in death. But the more important point is that, while during several hours the amount of glucose escaping from the killed loop diminishes progressively, The longer the penetration is allowed to proceed, the rate of absorption through the living loop remains practically constant over several hours. This same difference, shown by the surviving and the killed excised loop, appears under normal conditions in comparing the absorption from the peritoneal cavity on the one hand and from the intestinal cavity on the other. This constant shift of glucose, or more generally of certain hexaces, is the most conspicuous indication in intestinal absorption of transfer through some through-travital activity, as contrasted with passive penetration. It is convincingly demonstrated by showing that over a certain range of concentrations, about 6 to 12% with rats, the same amount of sugar leaves the intestinal fluid in the same period of time. This is evident, for example, from experiments of Verzar, which are listed in Figure 60. The average amounts of sugar absorbed within 1 hour are plotted against the amounts introduced into the loops in SCC of water. It is evident that glucose, A, and galactose, B, form one group, sullose, D, sorbose, C, and mannose, a second group, whereas fructose, C, takes an intermediary position corresponding to physical diffusion. The rate of absorption rises in the second group rather RCF-fedularly with rising concentration, whereas glucose and galactose are considered by Verzar to show a fairly constant absorption rate between 5.5 and 13%. The behavior of fructose approaches that of the second group. In the concentration interval of 2.5 to 5.5%, the absorption rate of glucose and galactose increases like those of the sugars of the second group. Below 2.5% within the 1 hour of duration of the experiments, the entire amount of these sugars present is absorbed. These results suggest the belief that the selective absorption of glucose and galactose is due to some kind of carrier system, which is seated in the intestinal wall and which is unable to shift more than the maximum load of these sugars at one time. Since it is unfit to carry the other sugars, it leaves them to an unselective physical diffusion. This hypothesis can be supported by various observations. On the basis of the well-known work of Lunsgaard on the severe alterations of carbohydrate metabolism by autoacetate Wilbrot and Last, have found that the preferential absorption of glucose and galactose is markedly impaired by this poison, so that the characteristic difference between these sugars and mannose, sorbose, and the pendices almost disappears, further postulating that phosphorylation as a regular intermediary reaction in carbohydrate metabolism may be involved in the preferential absorption of sugars. They have found that, on the one hand, addition of phosphate, pH 7, to the intestinal solution seems to speed the rate of absorption of glucose, not of acylose. On the other hand, extracts of intestinal mucosa, normally able to link inorganic phosphate with glucose, galactose, and fructose, but not mannose and xylose, lose this phosphorylizing power in the presence of iodoacetate, see later. Thus, phosphorylation may be connected somehow with the carrying function. About the different behavior of human erythrocytes towards sugars, see Sec 4, Chap 10. An especially strong argument for an intermediary phosphorylation is the action of another poison, fluorizin. This glucoside has been known for a long time to prevent reabsorption of glucose in the kidney. Its inhibitory effect is much milder than that of iodoacetate. This latter substance very easily brings about irreversible damage of many cell functions. It interferes with many transport reactions, for example, the secretion of diastopes by the kidney and the liver, the reabsorption of TL, and the absorption of amino acids. In general, these reactions are definitely stopped, and the effect is often manifested even by gross pathological changes. Furthermore, depresses in a fully reversible manner the intestinal absorption of glucose. It does not affect the absorption of glycine, glutamic acid, and asparagine. The effect of fluorizin is illustrated by the following experiment of Donley-Ofer. Under identical conditions, loops of the intestine of a rabbit are filled with equal volumes, 10 cc, of 2.5%, equal 250 milligrams, and of 15%, equal 1,500 milligrams. Glucose either with or without fluorizin. Of the 250 milligrams, Glucose present in the first loop, 123 milligrams, are absorbed in the absence of fluorescein, 19 milligrams. In presence of fluorescein, correspondingly of the 1500 milligrams. In the loop containing the stronger solution, 838 milligrams. Disappeared without fluorescein, 231 milligrams with fluorescein. This means that 123, 19 equals 104 milligrams. And 338 to 231 equals 107 milligrams. That is practically identical amounts. Are absorbed from the two loops through phosphorylation, whereas 19 and 231 milligrams, respectively, must pass the intestinal wall without phosphorylation, in other words, by syngenol diffusion. From the viewpoint of the carrier hypothesis, this can be interpreted as indicating that, irrespective of the great concentration difference, in the two solutions, the carrier is filled to the same maximum load, 104 to 107 milligrams, and that, in addition, 15.4% the amount absorbed from the weaker solution and 830% absorbed from the stronger solution pass by diffusion, but these conclusions should be substantiated by further study. Possible influence of two sugars administered simultaneously to the loop upon each other's absorption has also been investigated. Such a mutual influence has been found to exist in the case of glucose and galactose, which are absorbed from a mixture of equal parts of these sugars in such a way that the rate of absorption of each is reduced, so that the total amount absorbed is not greater than if glucose alone or galactose alone were absorbed. This, too, fits in with the idea of a carrier system which cannot transfer more than a maximum load. However, it has been found also that not only glycine and alanine, but also glycine and glucose influence each other in the same way. The opposite effect, then, with fluorescein, an ample increase of glucose absorption, has been recently observed with rats, which were given for several days thyroxine. Certainly, there is a number of factors which might be considered responsible for this effect, but depletion of carbohydrates, rise in the velocity of intestinal blood circulation, increased peristalsis, generally increased permeability of the mucosa, and others can be ruled out. On the other hand, evidence has been provided that the rats, after being treated with thyroxine, display a strong increase of absorption of glucose, galactose, oleic acid, that is, of substances which are readily phosphorylized, but fail to show the increase in presence of fluorescein, whereas thyroxine has no influence upon the absorption rate of alanine and of xylostat. Concerning the nature of the influence of phosphorylation upon absorption, more recent work has shown that this means much more than a simple intermediary reversible reaction with sugars, as in muscle metabolism. Phosphorylation is rather a sequence of interlocked transphosphorylations involving the intracellular action of a group of enzymes as mediators between the adenylate acid system and the carbohydrate system, and as such including an oxidative breakdowns of a fraction of the available sugar and thus a liberation of energy which is likely to be utilized for the transfer mechanism of at. This will be discussed in more detail later, chapter 38. Correspondingly, cyanide has been proved to inhibit the absorption of glucose. Regarding this situation, one may hesitate to use the term carrier systems to represent such a concatenation of chemical reactions. However, it will be shown later, P617, that the enzymes co-churned are localized in the body of the absorbing, e.g. epithelium, in a way as to suggest the functional participation of certain structural components, thus providing perhaps a vague idea about the histochemical nature of the transporting machinery. There is another mechanism that cooperates in the absorption of telephysiological sugars and is analogous to the conditions found to subpupil or the absorption of salts, pages 54. It has often been always survexed that glucose is absorbed along the concentration gradient from a higher concentration level in the ened system to a lower level in the blood. Rariny and Sperber have been able to demonstrate that glucose may also be shifted against the gradient by applying the principle of adding a slowly diffusible compound to the hypotonic intestinal sugar solution. as was found to be successful in demonstrating the marked uphill transfer of NaCl. For this purpose, they have used sodium sulfate or sorbose. After filling, for instance, a loop of a rabbit intestine with a blood isotonic solution of a mixture of glucose and sorbose, TGIE concentration of sorbose was observed to remain high throughout the experiment, more than 2 hours, while the glucose concentration rapidly dropped down towards zero, although the level of the rabbit's blood sugar was about 100 milligrams per cent for. The absorption of amino acids, since among the hexaces, the physiological sugars are subject to a preferential absorption, and among the salts, the most physiological one, NaCl, seems to undergo a corresponding treatment. It has been inferred that special cellular devices ensure a speedy absorption of the regular components of food. In this respect, particularly amino acids must be considered. The problem is simpler in the case of the hexaces, which are more nearly related to each other than are the amino acids, especially in the identity of their molecular volume. The amino acids possibly could be best compared to the acid amides, although the rate of their absorption has been shown to be inferior to that of the polyhydric alcohols, thus of a corresponding molecular volume, for reasons that so far are not clear. P. 535. Nevertheless, absorption experiments with amino acids appear promising for a problem in 2 respects. FD is conspicuous from old observations of overtime concerning the osmotic alveoluroproperties of frog muscle. That amino acids like glycine or alanine enter with great slowness, if at all, although because of their smaller molecular size, one could expect them to exceed, for instance, erythritol. Still more unexpected is their inertia in penetrating the cell surface of the sulfur alga vegetoa, which is remarkable for its outstanding behavior as an iono-killer sieve, Sec 4 chapter 10, and in which the permeation rates of several amino acids have been found to be abnormally low. The reason for this slowness is not of a physiological nature, since the slowness is evident also in diffusion experiments with collagen membranes, and can be explained as being due to the amphilite character of the amino acids, Sec 1 chap. 5, which probably due to the formation of a shell of water dipoles around the amphilite ions, brings about an enlargement of the molecular volume. Consequently, a porous membrane, such as the intestinal wall, CPO 534, would be expected to be passed comparatively slowly also. But the contrary is true. In comparison with acid amides, with erythritol, with xylose, the amino acids actually pass the intestinal wall much faster than was anticipated from their diffusion rates. Absorption of glucose, on the contrary, the absolute absorption within a certain concentration range, turns out to be fairly constant, and the percentage absorption decreases. See figure below, 547. Now, according to a series of preliminary experiments, the amino acids appear to resemble glucose. In table G5, the behavior of melanamide is compared to that of asparagine. These results concerning the absorption of amino acids are in agreement with others indicating an active reabsorption by the kidney 5, the absorption of water. So far, no conclusive evidence has been presented that aside from osmotic, colloidosmotic, and hydrostatic forces, page 270, there is a special driving force for water within the living intestinal membrane. Evidently, such a mechanism has been proved to exist within the excised surviving frog skin, transporting Ringer solution in one direction from outside to inside. P. 628. Rut the observation of a CX or throtting action of a diaphragm formed by a piece of fresh mucosa of a rabbit intestine as described by Reed could not be confirmed by other investigators. Corda paralo to the TISD8%, the kidney is like a very extended membrane of epithelial cells which separates the blood from fly urine. This membrane is arranged into a multitude of tubes, the nephrons. In the kidney, one end of each of these nephrons is closed by a Malpighian corpus L. All nephrons run practically side by side and end in a common collecting structure. All are supposed to be essentially identical as far as their anatomy and physiology are concerned. Our task in studying kidney function resembles the analysis of intestinal absorption, since it must be decided whether the efferniation of water and solutes across the membrane is a passive penetration or an active transfer. But the task here is much more diverse and thus more interesting than with the intestine, as each nephron is divided into a series of segments, each of them endowed with special functions, which mostly, but not always, appear to be indicated by a visible characteristic structure of the epithelial cells. We can distinguish as histophysiological units of each nephron the Bowman capsule in the Malpighian body, the J-roximal tubules, the distal tubules, perhaps better the proximal distal and the distal tubules, and in addition the thin segment of the loop of Henle, a special feature of the kidney of mammals. Thus, the contents of the nephron pass along the active epithelial wall like a technical product which moves on the assembly line, and which, by the addition of certain compounds and by the subtraction of others, is transformed into the final product, the urine. Thus, the kidney accomplishes its main function of eliminating the waste products of metabolism, which are circulating in the blood without abandoning valuable components which are on the way to get lost. After having entered the nephron through Bowman's capsule, the final goal is the maintenance of a normal internal medium surrounding the cells in the body and consisting of water and of certain solutes in proper concentrations. In view of such a goal, it is tempting to advocate a teleological point of view, and to foresee a selective and active transport for those substances that are either useful or useless to the body as a whole. Although many a recent finding is contradictory to this viewpoint, perhaps only owing to our meager ability to judge what is useful and what is not. The selective behavior of the intestine would FE likewise, in agreement with such a teleological concept, CP 550, on the other hand, taking into consideration the customary experimental procedure for examining the behavior of non-physiological foreign substances, for example, dyestuffs. It will appear that a certain correlation between the physical or physicochemical properties of these substances and the selectivity of their transport can be detected, which eventually may serve to eliminate the mystery of a teleological concept. For instance, a toxic foreign substance is expelled from the body by an active process in the kidney and, in addition, proves to possess such physicochemical properties as will precondition the release of the secretory mechanism in the analysis of kidney functions. It has, for a long time, been recognized as the primary task to attribute to the different portions of the nephrons the various functions, which become obvious even in a superficial view of the urine formation. Thus, merely on the basis of the histological picture, the glomeruli have been surmised to be a place of filtration. Further, a comparison of the various ratios of concentration in the blood fluid and in the urine has shown the tubules to control this interrelationship either by allowing a passive penetration or enabling an active transfer, the latter being either reabsorption of certain substances from the filtered fluid or secretion or excretion of certain other substances. 1. The formation of the glomerular fluid. Convincing proof has been furnished that the glomerular fluid is the result of filtration of blood fluid through the walls of the tuft of capillaries into the Bowman capsule. After numerous investigations, inaugurated by Carl Ludwig, 1844, it has been conclusively demonstrated that the rate of urine flow varies as the blood pressure in the renal arteries is raised and diminished without changing the amount of fluid passing through the kidneys, provided that the area of the filtering membrane does not change simultaneously due to changes of the blood distribution inside the kidney through local vasoconstriction or vasodilatation. Furthermore, the rate of urine flow decreases when the effective gradient of filtration pressure is diminished by raising the intrareteral pressure. The most direct approach for determining whether the glomerular fluid is a filtrate has been made possible by withdrawing fluid from the Bowman capsule as fast as it is formed. This procedure of puncturing the pathways of the nephrons, which has been introduced by A.N. Richards, permits the analysis of contents often amounting to not more than 0.5 cmn regarding capsule fluid of the amphibian kidney. The result is that glucose chloride urea creatinine, and hydrogen ions are found to be present in approximately the same concentration as in the plasma. But the fluid is protein-free. In other words, the capsular fluid can be looked upon as an ultrafiltrate. A corresponding condition has been found in the glomerular fluid of rat and guinea pig. Attempts have been made to obtain a more definite knowledge as to the physical properties of the glomerular membrane as an ultrafilter. After being injected into the blood of mammals, or after perfusion through the kidneys of amphibia, appear in the urine. The main result is that, grossly, the ability to penetrate the glomerular membrane is a matter of molecular weight. Hemocyanin 500, casein 190, serum globulin 140, serum albumin 680 are retained. Egg albumin 840, and Bence-Jones protein 840, gelatin 850, insulin 870 pass. Still greater is the permeability to PPL. Tuberculin 18500. Inulin 5100 has been found to appear in unchanged concentration in the capsular fluid. Dimethylamine 680 shows an ambiguous behavior, possibly due to the intermediary value of its molecular weight. Serum albumin is the first of the plasma proteins to appear in the urine in kidney disease. 2. The function of the kidney tubules. Fraud methods, the amphibian kidney, Fraudnecturus, has been found to be the most satisfactory object for analyzing the formation of urine. Not only does it display the common advantages of the organs of cold-blooded animals, which endure manipulation and isolation much better than mammalian organs, but it has specific anatomical advantages. The dual supply of blood from the aorta chiefly to the ventral half, where the major part of the glomeruli and of the distal tubules is located, and from the renal portal vein to the main site of the proximal dorsal tubules, has contributed largely to the development of kidney physiology. For owing to this twofold blood supply, it is more or less possible to provide separately the two halves of the kidney with different solutions, and thus to investigate the functional capacities of the proximal and the distal tubule. A practical procedure for such experiments with the isolated ringer perfused fraud kidney was proposed by Kulastai H.E. An important point is to keep the aortic and the portal pressures within the range of about 24-12 centimeters of water, corresponding approximately to the normal ratio of the pressures in the supplying vessels. Under these conditions, the arterial and the portal fluids, although both are confluent into common roots of the renal veins, usually remain sufficiently separated to allow a differential supply of the proximal and the distal tubular epithelium. This can be shown with diastuffs, the appearance of which, even in minute concentration, is easily detected in the secretion of 1. When the kidney is supplied under the normal pressure conditions from the aorta with ringer, from the portal vein with ringer plus diastuff in a low concentration, 0.1 milligrams percent. The diastuff appears in the urine in a remarkably enhanced concentration, 10 times and more, provided that it belongs to a certain group I, which, owing to a special molecular structure, see pages 563, has adequate properties. Under the same conditions, another group of diastuffs, too, fails to appear in the secretion, or it appears at best in an extremely low concentration, 5.1. If the kidney is perfused with ringer plus a diastuff I exclusively from the aorta, the perfusion from the portal vein being omitted, the dye appears in about the same increased concentration as in experiment 1b2. If this experiment 5.1 is repeated, substituting a diastuff 2 for a diastuff I, the dye appears either in the low concentration of the perfusion fluid or only slightly 2 to 4 times concentrated. This slight increase probably is due to water reabsorption. See pages 561 and 568. C. Supplying either a diastuff I or a diastuff 2 from both sides gives practically the same result as the experiments of 1 and a 2. From these facts, the following conclusions can be drawn. First, the proximal tubules are able to perform a secretory concentration of diastuffs I, not of diastuffs 2, experiments an I and a 2. Second, if, as in experiments B1 and B2, the portal pressure fails to balance the aortic pressure, the fluid entering the kidney from the aorta flows down to the capillary to the portal side, and thus circulates around the proximal tubules. Due to this connection between the aortic and the portal stream bed, the extent of which varies from individual to individual, eventually, but not necessarily, some fluid may pass over from the portal to the aortic side. Corresponding results have been gathered with colorless substances, for instance, similar to the group 2 of the dyestuffs, inorganic ions and organic non-electrolytes, even some belonging to the regular components of the plasma, may fail entirely to appear in the secretion when introduced from the portal vein only, the glomeruli at the same time being supplied with ringer. This has been found with phosphate, chloride, sulfate, thus. This simple method of dual perfusion of the amphibian kidney suffices to permit an extensive study of the correlation between the chemical and physicochemical behavior of solutes and the permeability and secretory capacity of the proximal tubules. Of course, only semi-quantitatively, but exactly enough to justify its often contested value. See also P568. A more direct approach to the problem of tubular function has been made by Richards and Walker-Dye in extending the method of glomerular puncture to the tubules of the amphibian kidney. Single tubules are punctured at various levels, and the contents collected and analyzed. For this purpose, certain segments can be isolated by blocking the lumen at 2 points with droplets of mercury or of colored mineral oil. Also, such a segment can be filled or perfused with a certain solution and re-emptied for analysis. Puncturing the Bowman capsule and the proximal and distal tubules at different points can provide information about changes of the composition along the nephrons. The results of this elegant procedure will be referred to later, as well as the use of OKT more simple methods, such as the study of the excised kidney explants and others. Beyond these methods, which subject the kidney to more or less urophysiological circumstances, other means have been found to explore tubular function in the normally blood-perfused kidney of the intact animal. The main procedure is based upon the determination of the renal clearance of the substances in question, see P574-5, passive penetration, in analyzing intestinal absorption, It has been found useful to ascertain to what extent the absorbing membrane can be looked on as being a diffusion membrane, and absorption not more than a passive penetration along concentration gradients, and how far special forces are engaged in bringing about an active transfer. The same procedure may be followed in dealing with the epithelial tubes of the kidney. But here we also are interested to know whether passive penetration across the membrane occurs in one direction as well as in the other. Penetration toward the lumen of the cephron. As mentioned before, a number of inorganic ions, phosphate, sulfate, ferrocyanide, chloride, when offered to the proximal tubules from the portal vein, cannot or can hardly pass into the lumen of the nephron. The behavior of chloride is of special interest. When in the arterial perfusion fluid, the normal Cl is replaced, remains Cl free. This is an especially striking demonstration of a unidirectional permeability of an epithelial layer, since Cl is moved to completeness in only one direction, CP559. But this Kl shift is a specific reaction of the chloride ion. Sulfate, phosphate, ferrocyanide probably are unable to leave the lumen after having entered the nephron through the glomerular membrane. They appear in the frog urine slightly concentrate due to water reabsorption, see pages 568. Many organic anions, especially the anions of sulfonic acid dyestuffs, and of related acids, show a corresponding incapacity to pass. About the passive penetration of inorganic cations across the tubular wall, very little is known as yet. The proper interpretation of this impermeability is that the molecular dimensions of the ions are too great to allow them to penetrate the pores of the civetite membrane. Such a view joint is suggested by corresponding experiments with organic non-electrolytes, likewise introduced from the tall side only. With polyhydric alcohols, it has been observed, that the rate of penetration across the proximal tubular walls seems to increase in the order. Mannitol glycerol, ethylene glycol, with aliphatic acid amides in the order. Malonamide acetamide, with sugars in the order silosa dioxyestin, in other words, in the order of decreasing molecular volume. This is similar to the situation in inactive penetration of the intestinal membrane, P538. The passivity of the processes is emphasized delocked by adding a toxic, but not disintegrating, substance. As in intestinal absorption, another factor seems to control the rate of passive penetration, namely lipoid solubility, CP. 535. There are not many observations which can be mentioned in favor of this correlation. Though having a molecular volume greater than silosa or arabinosa, enter the nediron, whereas the penises do not ought penetration from lumen to blood. So far as passive penetration in direction lumen to blood is concerned, very little information is available. Regarding the influence of molecular volume, it is interesting to note that with the technique of Richards and Walker, CP 557, proximal tubules of nectarus have been perfused with ringer containing urea in concentrations either higher or lower than that of the plasmodiode. From a merely physicochemical viewpoint, this exit toward the blood could be expected because urea has an exceedingly low molecular volume. In addition, other observations, CP 572, have suggested that urea leaves the tubules by passive penetration. However, it will be shown that urea, one of the most important compounds concerned in urine formation, exhibits an especially complicated behavior, CP 573. In the case of phosphate, also, passage in either direction obtains when single proximal tubules of nectarus are perfused. This is at variance with what was mentioned before regarding phosphates, R. Höber, unpublished experiments. C. Reabsorption by active transfer, reabsorption of inorganic ions, assuming that urine formation is begun by filtration in the Malpian bodies. The fact that normal frog urine is entirely or nearly Cl-free is the strongest evidence of active reabsorption of the filtered Cl against a steep concentration gradient into the Cl-rich plasma. And, since the normal urine of the frog is strongly hypotonic, one cannot evade the conclusion that simultaneously with Cl likewise is reabsorbed. This reabsorption in frog and in nectarus is a function of the distal tubules. In the proximal tubules, neither the Cl concentration nor the osmotic pressure of the fluid changes. But in the distal tubules, it drops progressively until it reaches 0 IBD. Reabsorption of Cl can also be demonstrated by perfusing the isolated frog kidney from both vessels with ringer. At the beginning of the experiment, the secretion frequently is found not to contain more than 0.1% NaCl, compared to the 0.65% of the ringer solution. But during the following hours, the concentration slowly rises and finally reaches 0.65%, indicating that the organ under the abnormal conditions is slowly dying. From a biological viewpoint, it is easy to understand that the heavy burden of doing continuous osmotic work is imposed upon the amphibian kidney to counterbalance the large absorption of water through the skin and its abundant output through the glomeruli, without loss of the substance that mainly contributes to osmoregulation, see Chap 36. On the other hand, as will be shown later, pages 561, 568, water also is likely to be reabsorbed in the tubules, and this may explain the fact that after the kidney has been poisoned, for example, by cyanide, a narcotic, or mercuric chloride, not only the Cl concentration in the urine rises, but also the amount of urine. All the more is it important to point out that active transfer of inorganic anions plus inorganic cations can take place without a concomitant. Previously it was mentioned. P 557. That sulfate, phosphate, ferrocyanide probably are unable to re-enter the amphibian body after having passed through the glomeruli. This would explain why effects similar to those observed with intestinal absorption are produced by these ions. According to Goldschmidt and Dayton, p. 542, from a mixture of sulfate and chloride introduced into an intestinal loop, the Cl shift into the body increases with rising sulfate concentration. In a similar way, when the isolated frog kidney is supplied alternately with ringer, and with ringer in which a part of Cl is replaced by sulfur, Again, it appears that the presence of sulfur favors the reabsorption of TL, obviously, in either case. Sulfate acts by its low diffusibility, P543. It is this factor which determines its diuretic and its diuretic effect if brought into action from inside the epithelial tubes. It further has been known for a long time that during idiuresis affected by sulfate, the percentage of Cl in the urine may drop to another inorganic anion, which is of particular interest because it helps to maintain the acid base. Balance in amphibia, as in mammals, is the bicarbonate ion. In perfusion exagerments with frogs, the PW of the perfusion fluid buffered by CO2 and elecos ordinarily is kept equal to 7.5, corresponding to the normal plasma value. In frog urine, the pH varies between 7 and 4.5. By omitting bicarbonate in the perfusion fluid, the PTI is brought down to about 4.8. The HEO3 content of acid urine frequently has been found to approach 0. For these reasons, it was suggestive to assume that the acidification of urine is based upon reabsorption of HEO3. The following experimental results seem to be in favor of this interpretation. By testing the redaction of the glomerular fluid with indicator diastuffs phenyl red fluorescein, or by direct electrometric determination, evidence is obtained that plasma and the glomerular fluid have the same pH and investigation of the PLL changes along the tubules. With phenyl red shows the acidification of the contents to be a function of the distal tubules, and to be restricted to a short segment near the distal end of the distal convolution. This marked localization in contrast to the more extended distribution of the reabsorbing power. For Cl indicates that two independent mechanisms are involved in the transport of these ions. However, so far no unambiguous evidence is obtained that bicarbonate is reabsorbed. Secretion of acid, likewise, must be taken into consideration. The following observations appear to be more in favor of the first interpretation, although they likewise give no conclusive evidence. By adding cidphenylamide and phenol red to the perfusion fluid, the normal acid reaction of the frog urine, as indicated by a yellow color, is changed to an alkaline reaction, pink color. This process is reversible. Besides sulfonylamide itself, derivatives containing the sulfonamide group unsubstituted have the same effect. Such compounds have been found by Mann and Keelan to be poisonous to carbonic anhydrase. This enzyme is a normal component of the kidney. Therefore, the bicarbonate in equilibrium with the bicarbonate in the perfusion fluid in passing the critical tubular segment is not unlikely normally to enter the wall and take part in the catalyzed acid reaction in the urine. Absorption of sugars, various observations had led to the assumption that the plasma glucose entering the pathways of the kidney 3% disappears from the secretion by tubular reabsorption before definite evidence was presented by GAA Lart, showing that from the glucose realm, perfused frog kidney glucose is no more retained. After the proximal tubules have been poisoned with mercuric chloride by way of the portal vein and glomerular membrane into the Bowman capsule in unchanged concentration. The site at which the reabsorption takes place is detected by puncturing the tubules at various places. It appears that, in contrast to the behavior of Cl, it is in the proximal tubules that the percentage of glucose falls off to a value near 0 GDI. Moreover, it appears that the air caps of glucose can be entirely suppressed by J-Lord. Their IFDC, or glucose concentration, increases along the nephron until at the end of the proximal tubule, it is about 40% above the plasma level. At the end of the distal, about 200%. This probably is due to reabsorption of water occurring particularly in the distal, and is comparable to the increase of concentration observed with inorganic and organic ions. Pages 556, 558. Other sugars besides glucose, hexases, as well as pentoses were studied at first by H.J. Hamburger by perfusing the frog kidney from the aorta only, so that the perfusing fluid reached more or less the entire length of the tubules. CP 556. Under these conditions, the sugars appear to be retained to various degrees. Clearer results are secretary by perfusing alternately with two sugars, either from the aorta alone or from both supply routes, and checking simultaneously the kidney function by CL analyses of the urine and by determining the secretory concentration of phenol red continuously injected through the portal vein. In this manner, the following order of absorption rates has been found: glucose, galactose, mannose, fructose, dextrose, arabinose. In other words, the sugars are arranged in an order which does not differ very much from that encountered in intestinal absorption, P546. As a matter of fact, the two absorption phenomena resemble each other in more than one respect. First, in both cases, we find the poisoning action of fluorescein, and this effect is found to be stronger the higher the absorption rate of the sugar. This correlation was detected independently in the kidney experiments and in the study of Lunsgaard and of Wertheimer on intestinal absorption, P548. Second, the amino acid glycine undergoing a selective absorption in the intestine, as well as in the kidney tubules, CP550, is equally well absorbed, no matter whether fluorescein is present or not. P563. Also, the absorption of Cl or the secretion of phenol red is not depressed by fluorescein. Therefore, the conclusion may be drawn that, as in intestinal absorption, the sugar absorption is tied up with phosphorylation. Now, since enzymatic phosphorylation is dependent upon a supply of oxygen, P550, and can be suppressed by poisoning with cyanide, sugar absorption also can be conceived of as being an active transfer. The following experiments are in favor of this viewpoint. The ringer perfus, frog kidney is supplied from the aorta with glucose, from the portal vein with phenol red, and in addition for a short time, with phenylurethain as a metabolic poison, CP869. The result is that temporarily during the narcotic period, the glucose concentration in the secretion increases, the phenol red concentration decreases. Since either of these processes depends upon activity of the J-roxymal tubules, or if the effect of the narcotic were nothing else than an increase of permeability of the tubular wall, the concentration of glucose would not be expected to rise. Therefore, if in another experiment, the kidney is perfused from both sides with glucose ringer and brings about by its reabsorbing power a continual shift of the sugar against an increasing concentration gradient, the rise of glucose concentration, reversibly accomplished by phenylurethain, cannot mean anything else than active transfer. In other words, osmotic W-ORC. See, however, again similar to the conditions in intestinal absorption of glucose absorption, P58. The transfer toward the blood by the activity of the tubular epithelia is raised by the administration of thyroxine, and as there are reasons to believe that the increase of absorption is not due to a more abundant blood circulation, to an enlargement of the active mass of cells, to a rise in temperature, etc., and as further the increase can be abolished, phosphorylation. But this interpretation needs more discussion, since it has been discovered that fluorescein is able to retard the active transfer of substances which are very unlikely to be phosphorylized. See pages 572 and 7a, absorption of amino acids. So far, there is not much known about the ways and means enabling the amino acids to pass the amphibian kidney. This is chiefly due to the fact that difficulties were encountered in determining exactly, by the anhydride reaction, the small amounts of these substances, which appear in the secretion. The amino acids seem to resemble glucose in their behavior, viz. Glycine, alanine, tyrosine, tryptophan, supplied from the portal vein, do not enter the tubules, whereas after they have been supplied by both routes, the secretion does not contain more than 3/2 of the perfusion concentration, the major fraction being absorbed. Further, after inactivating the proximal tubules by a narcotic, the reabsorption is abolished. On the other hand, fluorescein fails to inhibit the absorption of glycine in the kidney, as it fails to do in the intestine, P548. Corresponding results have been obtained with dogs, haptidine. Secretion by active transfer. The word secretion will be used in the following paragraph as indicating active transfer in the direction blood to lumen. Secretion is usually associated with increase of concentration, but not necessarily. The analogous term used in the preceding paragraph is reabsorption. It indicated active transfer in the direction lumen to blood, and as yet this active transfer seems to be confined to valuable substances, such as sugar, amino acids, salts, which serve in the body as foodstuffs or for osmoregulatory or other specific purposes. Whether this biological interpretation of the function, which was suggested by the study of intestinal absorption, CP 550, may be generalized, can be decided by determining whether useless substances, for instance, waste products of metabolism, are also actively reabsorbed in the kidney. It is true, Chirea appears to re-enter the body in the mammalian kidney, but probably by passive penetration, p. 573, however, in the kidney of elasmo branch, where urea has to fulfill a special osmoregulatory function, it undergoes active absorption, p. 573, on the other hand. Not only compounds such as those produced as metabolic end products can be subject to active secretion, possibly owing to a special adaptation of the kidney cells, but numerous foreign substances also have been found to set going the secretory machinery. How this can take place is one of the main problems of the following paragraph. The secretion of dyestuffs, previously, p. 556. It was explained how dyestuffs can be utilized not only to trace the pathways of dissolved substances through the amphibian kidney, but also to provide information about the functional differentiation of the main segments of the nephron under various conditions of perfusion. In other words, by measuring the accumulation ratios. However, it must be added that here, as well as in the study of other secretory functions, it is advisable to employ acidic dyestuffs instead of basic ones. Basic dyestuffs are the classical vital stains of Paul Ehrlich, which, due partially to their basic character, partially to their lipoid solubility, are admitted to more or less all living cells, whereas certain acidic dyestuffs, especially certain sulfonic acid dyestuffs, are selected by special cells for active transfer or active uptake and cannot enter after inactivation of these cells, CEG, pages 670. To this latter category of cells belongs the tubular epithelium of the kidney. There, the appropriate acidic dyestuffs, while being transported in general, display a homogeneous, rather faint coloration of the entire protoplasm. By the earlier investigations, the effective perfusion of the kidney with a dyestuff often was described as it appeared under the microscope in slices of the excised organ. In such preparations, the cells contain deeply colored vacuoles, which are mostly artifacts, arising in the dying protoplasm, especially after an excessive infusion of dye. On the basis of these observations, the long and unproductive discussion, referring to the alternative secretion reabsorption, has taken place. It also has been mentioned previously, p. 556, that the dual dyestuff perfusion does not allow a clear-cut differentiation between proximal and distal functions. Nevertheless, at least per exclusion, it can be shown that secretory activity mainly is due to the proximal tubules. If one of those dyestuffs, which can be concentrated by the kidney, is introduced from the aorta only, the portal vein being supplied with ringer, the secretion will be slightly colored, not more, or at best little more than the perfusion fluid, although the aortic perfusion not only supplies the glomeruli, but spreads around the distal tubules. But the reverse arrangement, aorta ringer, portal vein dyestuff ringer, conveying the dye to the capillaries around the proximal tubules, is followed by the formation of a markedly colored fluid. However, the color intensity under these conditions is found to vary greatly, among other reasons, because the amount of glomerular fluid which mixes with the proximal secretion is a variable factor. The following three experimental objects are free from this complication, and therefore can provide somewhat more direct evidence of the tubular activity. The aglomerular kidney, p. 568, the excised surviving frog kidney, p. 567, and the kidney explants of the chicken embryo, p. 569. The secretion of the sulfonic acid dyestuffs, the first definite proof of a secretory activity of the kidney tubules, was given by E.K. Marshall. J.R. 114 showed that after a subcutaneous injection of phenol red, the amount of dyestuff available in the blood for filtration in the glomeruli at a normal rate of blood circulation is too small to account for the amount of dyestuff accumulated in the urine. The conclusion was drawn that the dyestuff concentration in the urine could not be due to reabsorption of water alone. More directly, the dyestuff can be proved to be transported through the proximal tubules by the method of the dual perfusion with Ringer. Upon comparison of a great number of readily diffusible sulfonic acid dyestuffs in low concentration, for example, 0.005, it is evident that some of the technical colors in aqueous solution include all degrees of dispersion from molecular of the dyes are concentrated by the iroximal tubules to a greater or smaller extent, and others are not. This is especially conspicuous when from a mixture of one specimen of each of these groups of dyestuffs being tested. A more or less complete the mixture is brought about by the kidney, see pages 567, 568. In order to answer the question as to what factors are involved in such different behavior among technical acid dyestuffs, those belonging to certain chemical groups have been selected, chiefly azo, diazo, triphenylmethane, and sulfonaphthalane compounds. Among these, the study of the azo dyestuffs was found to be most enlightening. The azo dyestuffs, the basic structure of the azo dyestuffs is composed of two ring systems, either benzene or iophthalane, linked together by an azo group. To this skeleton, there are attached one or more sulfonate groups as well as other radicals, NH2,2C3 and others. The greatest bearing upon the physiological behavior of these dyestuffs appears to be referable to the sulfonate groups, as indicated by the following observations. 1,10-benzene-azo-naphthalane monosulfonates were studied. They appeared accumulated in the secretory, no matter whether the sulfonate was on the benzene or the naphthalene ring. With 8-benzeazo-naphthalene disulfonates, the essential point is whether one of the two ring systems carries both sulfonates or each ring system carries one of them. In the first case, the dyestuffs, 5, were accumulated by the kidney. In the second case, 8, they were not. 3. A similar behavior is shown by naphthalane-isonaphthalane disulfonates. With 5 dyes, the attachment of both sulfonates to one of the naphthalanes resulted in a marked accumulation, while 4 out of 5, with one sulfonate on each side, were concentrated only very weakly. 4. The result with trisulfonates is irregular distribution to colloidal solution of the hydrophobic or the hydrophilic type, CSX5, CHAP-EFA. Correspondingly, after an intravenous injection in ample concentration, some dyes pass the glomeruli, others do not. Some appear more quickly, some more slowly in the secretion. In general, the semicolloidal and colloidal dyes, in contrast to those in molecular dispersion, are deposited after a time in the form of granules in the proximal epithelia, first in the vicinity of the glomeruli, then farther along the tubules, some for a shorter, others for a longer period. This is not a reabsorption in the usual sense, and is independent of the essential activity of the kidney. R. Elosier, Bio, Hampshire 20. Gerard and R. Cordier Hyol, Reverend 9, 110, 1934. See further pages 615. The following interpretation has been proposed. The molecules of the azo dyestuffs are characterized by a bilateral and possibly elongated structure. Those with the sulfonate groups on side have the character of a dipole, the non-sulfonated half being non-gene-olar and organophilic hydrophobic, the sulfonated half iolar and hydrogylic, see chap 20. According to the constitutes developed by Langmuir and Harkins, the dipole molecules can be assumed to be oriented in such a way as to bring about a loose fixation of the organophilic half to the cell surface, whereas the hydrophilic half is turned toward the water. The dyestuffs with hydrophilic groups on either side may be supposed to lack such an orientation and fixation. Eventually, the influence of other radicals besides sulfonates, displaying a lesser affinity toward the organic surface, can invalidate the hydrophilic forces. The attachment to the cell would be conceived as being the first step toward the entrance into and the transfer across the cell. However, Another factor favoring the entrance of the dyestuffs has to be discussed: lipoid solubility. Previously, it was shown that the term lipoid solubility is somewhat misleading, insofar as it pretends to refer to a simple solution in an organic solvent. However, lipoids are mixtures of organic compounds, which, at least partially by chemical affinities, affect the distribution of organic substances. Now, many of the azodiestuffs have been found to distribute between water and a lipoid, quite shaken with both solvents, employing as a lipoid phase a mixture of oil plus oleic acid plus diamylamine, see Chap 13. For example, all benzeneazonaphthalene monosulfonates largely enter this mixture from aqueous solution, indicating that the organophilic affinity of the ring system is strong enough to overcome the hydroaffinity of the one sulfonate group. However, among the benzeneazonaphthalene and cholinaphthalene-azonaphthalene disulfonates, there are compounds combining a distinctly nonpolar-polar configuration with a live accumulation, some of them being lipoid-soluble, others lipoid-insoluble. Therefore, lipoid solubility cannot be conceived to be more than a secondary factor in their secretory transfer, the triphenylmethane dyestuffs. Similar considerations seem to be serviceable in accounting for the conditions encountered in the secretion of the triphenylmethane dyestuffs. The basic structure of these dyestuffs, a central C atom surrounded by 3 benzene nuclei, does not lend support to the idea of the necessity of a bilateral configuration. Further, all sulfonic acid triphenylenethanes studied so far, 14 specimens, are lipoid insoluble. It may be due to these properties that the ringer perfused fraud kidney either fails to respond to the presence of the triphenylmethanes, or does so less than usually observed the conditions of a perfusion experiment. Whereas the portal J fusion with the solution of an appropriate azodyestuff as weak as 0.1 to 0.5 milligrams. Percent P564 is succeeded by an accumulation in the excretion in the corresponding experiments with the triphenylmethanes. The perfusion concentration has to be raised to a level 10 times higher or more in order to cause the appearance of dyestuffs and even a IOIV concentration. And this only if the perfused dyestuff possesses an aromatic amine in its molecular structure. These observations suggest that chemical affinities are involved in the secretory uptake, in place of the nonpolar organophilic affinities of the azodyestuffs. See further P570. Finally, it should mention that there is no better method to differentiate between secretory concentration and concentration by reabsorption of water in kidney function than to make use of the diverse qualifications of the sulfonic acid dyestuffs for secretory transfer by perfusing a mixture of 2 dyestuffs which differ distinctly from each other. For instance, when a frog kidney is perfused from the arterial and from the venous side with Re-Ike plus 0.8 milligrams percent of the lipoid insoluble triphenylmethane, cyanol, the secretion shows a trace of blue color corresponding to a 2-fold concentration, due mainly to reabsorption of water. In a second period of the experiment, with perfusion again from both sides with 0.3 milligrams percent cyanol, in addition to 0.3 milligrams percent azofucine first, a lipoid insoluble secretable azodyestuff of red color. The secretion shows an intensely pinkish red color corresponding to a 20-fold concentration of the azofucsine. If only reabsorption of water had taken place, both dyestuffs provided at the same concentration to either side of the epithelial wall should have been concentrated to the same level. It is obvious that the separation of the 2 diffusible dyestuffs is the result of their different kidney function without glomeruli. The experiments with azo and triphenylmethane dyestuffs concerning the secretory properties of the proximal tubules are supplemented and partially corroborated by the observation of preparations, in which the function of the glomeruli is more or less excluded. The excised kidney. Richards and Barnwell-Falk have shown that the excised fraud kidney, immersed in well-aerated ringer, retains its activity for some time. This is indicated by the facts that, first, after 0.01 to 0.3% phenyl red has been added to the ringer solution, dark red threads appear under the microscope in the lumina of the tubules. Second, addition of amyl cyanide prevents the accumulation of the dye. Extending these observations to other dyestuffs, it is tendency to show that the dyestuffs in general behave just as to be expected from the perfusion experiments, provided that one takes into account that instead of oxygen distributed by the blood vessels throughout the entire organ, the dyestuffs enter the excised kidney from the surroundings by diffusion across its surface, and that, therefore, whatsoever changes may be brought about in the composition of the tubular contents by the activity of their walls will not be nullified. The microscopic pictures ordinarily AI pairing after exposure of the excised kidney to one of the secretable azo-D stuffs is that on the dorsal side, the lumina appear to be filled with more or less intensely colored fluid, contrasting with the relatively pale epithelial wall, whereas on the ventral side, the tubules as well as the glomeruli mostly remain colorless. But this pattern never becomes visible when, after an ordinary perfusion with dye stuff in low concentration, the kidney is excised and examined under the microscope, even when the kidney had delivered a secretion showing a high accumulation ratio. Dye stuff solution, even at such a concentration level, is too high in the thin lernita to become conspicuous under the microscope. Any other factor contributing to the distinctness of the microscopic picture in the excised kidney is that the tubular contents not only fail to be dilux by the glomerular fluid, but, on the contrary, are reduced in volume by water. Hershey absorption. It becomes clear that even those triphenylmethane dyes, which were mentioned as appearing in the secretion during a perfusion experiment at best, at a very low concentration, can produce an excellent dyestuff pattern in an excised kidney immersed in the 0.03% solution of a suitable triphenylmethane, for example, cyanol or on the basis of other observations. The conformity of the action of the PC or fused in the excised kidney is immediately obvious. Thus, exposing one kidney to a desulfonated azo dyestuff with the sulfonate groups attached to one side of the molecule, for example, azofuccinate ars, the other kidney to the corresponding dye with the sulfonates opposing each other, azofuccinate G, the characteristic pattern appears with the first and does not with the second. Further, the excised kidney, immersed in the violet mixture of blue cyanol and red azofucin G, separates the two dyes so that a blue secretion becomes visible in the lumasa. But no pattern appears in the presence of a narcotic, for example, phenylurethanine or phenylura, be the glomerular kidney. Nature has provided the opportunity to study some fish with kidneys lacking or nearly lacking in glomeruli. Attention of physiologists has been turned to this material mainly by Marshall and Graflin and by Edwards. The chief species investigated have been the goosefish Lophius piscatorius and the toadfish Dopsanus tau. Their kidneys are composed of blind tubes, the walls of which arc formed by epithelial cells, with K-brush border resembling the proximal tubules of the glomerular kidney of other vertebrates, even anticipating the permeability of these tubes towards solutes to be like that of the proximal tubules of the frog or any other animal. There remains the special problem of water transport. Since the aortic pressure has been found lower than the secretory pressure in the ureter, the study of the passage of solutes gave the following results. After intramuscular injection, many substances appeared in the urine of the toadfish: uric acid, creatinine, iodide, the sulfonic acid dyestuff indigo carmine, and phenol red, the basic dyestuff neutral red, and others even more instructive are quantitative determinations. Ferrocyanide, silosa, glucose do not appear, whereas they are excreted in the urine of fish with glomerular kidneys. Previously, Year 557, it has been mentioned that the substances also failed to pass the proximal wall of frog tubules in the direction blood to lumen. 2. The glomerular kidney Lophius differs from glomerular kidney in being able to concentrate energy and so forth 30 times, and even Arnor taunt 3, provided the toadfish is handled very carefully, ideally avoiding any skin injury. The urine is practically Cl-free, CP. 585-4 over a wide range of Asheville's in the plasma. The concentration in the urine is found to be the same as in the plasma. This seems to indicate diffusion, probably due to the small molecular volume. See, further, P57-2. 5. The toadfish kidru is able to concentrate phenol red up to 150 times. Further, after the subcutaneous injection of certain diffusible sulfonic acid, azodiestuffs, the same microscopic picture appears as is seen with excised frog kidney. The lumina of numerous tubes are filled with color, the surrounding epithelial walls contrasting in their paleness. See explants of the mesonephros of chick embryos. Chambershaws discovered an excellent procedure to study the function of isolated tubules. The mesonephros of an 8 to 10 day chick embryo is cut to pieces. Tubules are mechanically separated from their glomeruli, and tubular fragments are explanted. In growing, the broken ends often become closed and are converted into cysts by the intake of fluid. Proximal and distal pieces are identified by their characteristic epithelial structure, by their greater dissension due to a larger fluid intake, and especially by the fact that only proximal cysts, while embedded in a very dilute solution of phenol red, take up the solution and transmit it into the lumen with a 30 times higher concentration. If in a cyst of a distal segment, incidentally, a bit of proximal epithelium is included, phenol red will stain only the perceivable cells, which are thus sharply differentiated from the colorless distals. The color appears in the cells in homogeneous solution, except under abnormal conditions, when cytoplasmic vacuoles are formed, CP16. Irrespective of the higher concentration of dyestuff in the cysts, the total molarity of the enclosed fluid, withdrawn from the cyst by a micropipette, in other words, its osmotic pressure has been found to be about 10% lower than that of the surrounding fluid. Its hydrostatic pressure, that is, its secretion pressure, higher. This latter fact corresponds to the aforementioned measurements of Beater regarding the glomerular kidney of the toadfish P569. Further properties of these preparations will be referred to lat, or see Chap 38. Secretion of non-dyestuffs. Dyestuffs, more than any other compounds, have proved to be appropriate for utilizing the anhydrous kidney to show the functional differentiation of the various segments of a nephron by inspecting the pathways of the color and measuring the colors. Strength. The sulfonic acid azodiestuffs in particular have provided some inkling as to a correlation between the molecular configuration of a dye and the secretory capacity of the proximal tubules, Chinese 565. These studies suggested investigation of simpler colorless chemical compounds by applying the following methods. Breaking the azo bond in an azo dyestuff yields two colorless halves, each of which contains a benzene or a naphthalene ring, and at least oik of them a sulfonate radical. The sulfonated half evidently has a nonpolar organophilic hydrophilic composition, Chap 20. The question whether as such it may potentiate a secretory transfer can be solved by determining whether or not the colorless secretion yields a color after another suitable half molecule of an azo dyestuff has been coupled in a diazo reaction to the substance in question. The intensity of the color eventually appearing in the secretion is indicative of the accumulation ratio, with a fairly great number of amino and hydroxynaphthalene monoandisulfonates supplied to the kidney from the renal portal vein. The simple result of this procedure has been that the mononaphthalene sulfon 8s undergoes secretory concentration by the proximal tubules. The disulfonates do not. Further, the aminobenzene monosulfonates also fail to be concentrated. Whether these findings may be interpreted as being due to the physicochemical character of a nonpolar-polar configuration is uncertain. Probably the result depends upon a balance in the molecules between the hydrophilic forces of the sulfonate. On one hand, and the organophilic affinity of a simple benzene or a double naphthalene ring, combined with chemical affinities of the various radicals attached to the ring system, on the other hand. This concept can be substantiated by the following observations. 1. Certain derivatives of sulfanilamide Nilt 1:1, which as such passes the kidney without being accumulated, can be converted into organophilic hydrophilic substances either by substituting for the sulfonamide group, a sulfonate, for instance, or a carboxyl group, or by attaching to the NH2, a fatty acid radical, for instance. They are then capable of being concentrated, too, on the background of the experience that an organophilic hydrophilic structure is one of the essential factors in kidney activity. New light is thrown upon the processes of physiological detoxification. Toxic substances, either arising as metabolic products or somehow entering the body, often are weak electrolytes and are soluble in lipoids, and for these reasons are apt to enter the cells. One customary procedure of the living animal for detoxification of these substances is their conjugation with relatively strong acids, like sulfuric acid, glucuronic acid, and others. This incorporation of the poison into a strong electrolyte in general results in incapacity to penetrate the cells. A second effect is the formation of a hydrophobic hydrophilic compound, which can be eliminated by way of the kidney cells, indoxyl sulfonate. But this conjugation also occurs with weaker acids, the strength of which is increased by the conjugation so much that again the product is highly ionized and acquires an organophilic hydrophilic configuration. Fg, the toxic benzoic acid is conjugated with glycine by coupling the benzoyl radical to the NH2 group. The conjugation product, hippuric acid, is a fairly strong electrolyte, K equals deuterium 10, and has an organophilic hydrophilic structure. Possibly for this reason, hippurane, that is O-iode hippurate, which is one of the most satisfactory contrast media for röntgenography of the kidney, is found to be eliminated by secretion. Still more applied in X-ray testing of kidney function is diodrast 3.5 acetate, where skiodine, another organic iodine compound, monoiodinothane sulfonate, fails to be accumulated in the urine. From the structural formulae, one possibly can conclude that this differential behavior is indicative of a correspondingly stronger or weaker polar structure of the molecules. Diodrast, furthermore, has been utilized for approaching an important physiological problem, viz., in how far the active reabsorptive transfer and the active secretory transfer can be paralleled. By the experimental investigation of this problem, new aspects of the transfer mechanism have been aroused. For reasons previously mentioned, glucose absorption in the intestine and glucose reabsorption in the proximal tubules arc assumed to need an intermediary phosphorylation. This concept is especially strongly supported by the inhibitory influence of fluorescein, pages 548 and 561. Now, as has been shown by White and by Feiler, Althausen, and Stockholm, both the reabsorptivity of glucose and the secretion of diodrast are diminished by fluorescein. But it is highly improbable that phosphorylation is directly involved in the transfer of diodrast, and another way of interpretation has to be looked for. This will be discussed later, P617. Urea and uric acid, attention is now turned to the output of two metabolic waste products of special physiological significance. In their analysis, great difficulties have been encountered, partly due to divergences in behavior from genus. Again, the main material for study was the amphibian kidney. Other investigations will be mentioned later. CP 578. Urea is one of the main organic components of the amphibian urine. Its concentration usually surpasses that of the plasma. The percentage in the contents of Bowman's capsule and the plasma have been found practically equal, but from the capsule downward, the concentration increases along the proximal and continues to rise along the distal tubule. Thus, the final concentration reaches in nectarus an average value of 2.2, in frog of 7.8 times the plasma concentration. In order to determine whether these differences are attributable to reabsorption of water or to secretion, fluoresceinized animals were used, with the result that in nectarus as well as in frogs, the average concentration for glucose was found to be 2.2 times the plasma concentration. That is, the same values as found for urea in nectar. Therefore, the conclusion is drawn that accumulation of urea in nectaris may be referred to absorption of water, but in frogs to both water absorption and secretion. This conforms to earlier experiments of Marshall comparing urea with the xylose, the latter being practically indifferent to this. In another way, evidence is afforded for an active transfer of urea by the frog kidney. Comparing the various urea concentrations in plasma to those in urine, it appears that the accumulation ratio in general is in an inverse relationship to the plasma concentration. The same phenomenon has been observed with dyestuffs, for example, phenyl red and other sulfophosphate, and in studies upon other kinds of secretory transfer, for example, plant cells, see Chap 36. And as such, the phenomenon is an argument for assuming active transfer, which is possibly effectuated by a carrier system, in concluding, it should be made clear that this important metabolite urea is subject to most variable modes of distribution. As just mentioned, among the amphibia, nectaris, and frog show great differences. In moths and in some teleosts, absorption, possibly only as a passive penetration in the direction of lumen blood, is a preponderant factor chi, also P579. And in Alasmobragics, dogfish, urea is actively reabsorbed by the tubules, apparently in order to participate in the osmotic balance of the blood of these animals, which has been known for a long time to contain 2 to 3% urea, besides a percentage of salt considerably lower than that present in seawater. Finally, in the frog kidney, urea can be stored to such an extent that its concentration at the dorsal side of the kidney, where the proximal tubules are located, by far surpasses the concentration in blood and in urine. The nature of the mechanism accounting for the storage is unknown. Uric acid. In order to study the elimination of urea to 3 milligram kidney, 1 to 3 milligrams percent uric acid as lithium urate are supplied to the isolated ringer perfused frog kidney either from the arterial or from the venous side. In the first case, it appears in the secretion to 2.5 times concentrated. In the second case, the average accumulation ratio is 10. In the light of previous considerations, P564, this is indicative of a secretory activity of the proximal tubules. This belief is supported, first, by demonstrating that M1000 cyanide, simultaneously administered through the portal vein, reversibly depresses the concentration and the amount of urate delivered. Second, by determining with a suitable dyestuff, cyanol, CP 566, that the supply of the urate is mainly confined to the proximal tubules. A similar situation has been found in lizards after poisoning with fluorescein. CP 561. The percentage of glucose and uric acid in plasma and in urine is compared. The average concentration ratio for glucose is found to be 3.2, for uric acid 51.4. Absorption of glucose being stopped centrated 3.2 times by tubular reabsorption of water. It follows that by far the most of the uric acid must be eliminated by tubular secretion. The fact that in man uric acid is believed to undergo reabsorption will be, again, information is needed as to whether the secretability of uric acid can be assigned to any of its chemical or physicochemical properties. As a matter of fact, uric acid and its salts are exceptional in that they have a tendency to form colloidal solutions, which readily turn to unstable ultramicroscopic and microscopic suspensions. As such, they may be stored. At least in frog, after an intravenous injection, there appear very fine precipitations solely in the cells of the proximal tubules, not in the distal tubules. The plasma clearance in Teodar, Effie, and Kuhn, thought so far chiefly, such experiments have been reported as were intended to elucidate the kidney function by subjecting this organ to rather artificial and syringe-defied conditions compatible with the living state of the organs of amphibia, that is, with ringer solution. Now we turn to experiments upon the kidney in its normal functional relationship to all the other organs, especially experiments upon the mammalian kidney. Certainly, this procedure is fraught with essentially greater technical difficulties, and in order to approach the goal of substituting the more qualitative results of the so far described experiments with more quantitative ones, It is necessary to keep the animal under certain basic conditions. The plasma clearance is the capacity of the kidney to clear during 1 minute a certain volume of the blood plasma, measured in cc, of an excretable substance. The amount of this substance is pxc. If P is its concentration in the plasma, see the plasma volume passing through the C for the following. H.W. Smith, The Physiology of the Kidney. It is referred to plasma and not to whole blood, since it is only the plasma which undergoes filtration, or rather ultrafiltration, in the nephrons, and since, due to the great variations in the distribution of the substances in question between the blood corpuscles and the plasma, the concentrations in the whole blood can be far from identical with the concentrations in the plasma. Glomerular membranes into the nephrons. The same amount of the substance reappears within 1 minute in the urine, provided that in passing the nephrons, nothing is withdrawn by reabsorption, and nothing is added by secretion. This amount in the urine equals UXK, if U is its concentration, and V the volume of the urine excreted. It follows that UV equals PC or C equals UVP, where under the conditions mentioned, C, irrespective of the plasma concentration, has been found to be constant. That is, the circulatory supply of the kidney is constant. Now, if the concentration of an excretable substance, after having passed through the glomeruli, is augmented by the secretory activity of the tubular walls, the constant C, as calculated from the above equation, appears to be increased. If it is diminished by the reabsorptive power of the walls, it is decreased. Thus, a way is open for deciding whether a substance passing the kidney, apart from being filtered, undergoes secretion or reabsorption. By comparing the substance with a sterile hard substance which is neither secreted nor absorbed, and which is best supplied simultaneously with the substance in question, probably there exist many substances suitable for such standards. They should comply with certain requirements. For instance, they should be chemically, physicochemically, and physiologically rather inert. They should have a medium-sized molecule and thus be filterable. And others, according to A&L. Kitchards and H.W. Smith, the starch-like polysaccharide inulin has been proved adequate. Suitable properties have been found also in mannitol and sorbitol further, in fluorescentized animals. In other words, in animals with kidneys disabled for reabsorbing sugars. P. 561, glucose, thiacylose, and sucrose behave like inulin. All these substances, when tested with the same animal, have led to fairly identical clearance values, irrespective of variation of the plasma concentration over a fairly wide range. An example using a normal humanis given in the following table. It appears that with moderately rising plasma concentrations of inulin 159 Smith, The Physiology of the Kidney, Oxford University Press, New York, 1937. P. U rises and V falls, evidently due to water absorption, but C, the clearance value is fairly constant, average 127 cc's. However, consistent results cannot be obtained in clearance determinations unless a certain basal status of the animal is maintained carefully. This means that, during each single experimental period of urine collection, the plasma concentration should be kept constant. Further, that changes of the blood pressure and of the rate of blood supply to the kidney, changes of the water balance and of the composition of the food, should be as to the effect of a secretory transfer upon the clearance values to results obtained in an experiment with phenol red and inulin simultaneously injected into a man, are outlined in Figure 61. The phenol red clearance, compared to the inulin clearance, the horizontal broken line in Figure 61 in terms of phenol red inulin clearance ratios, surpasses the inulin value equal to 1 by a maximum of 3.5 times. But it is not constant, as is the inulin clearance, see Table 46. Rather, it falls off above P equals 1 milligrams percent, and with P values of 10 to 20 milligrams percent, approaches the inulin clearance figure. This indicates that inulin and phenol red enter the nephrons through the glomeruli at the same ratio as in the plasma, but the percentage elimination of Phenol red by the kidney decreases when its absolute concentration in the blood rises beyond 1 mg%. Apparently, above this concentration limit, the mechanism of the secretory transfer has reached its maximum capacity. And an additional output after raising the phenol red concentration in the plasma has no other source than the amounts filtered through the glomeruli, which are in linear relationship to the blood concentration. This behavior is illustrated by figure 62, referring to the phenol red concentration by the frog kidney. The total amounts of phenol red excreted per unit of time are plotted against its plasma concentration, curve of dark circles. These amounts are equal to the product UV, P575. Their origin is partially glomerular filtration, partially PLG. The effect of plasma phenol red concentration on the amount of dye excreted by the tubules and the glomeruli of the frog kidney. Tubular secretion, the amount supplied by filtration at different plasma concentrations, broken line, can be calculated by applying the equation WIP equals C. Hence it becomes clear that, with rising P, the part of the total excretion which is due to filtration increases, whereas the part representing the transfer by the tubules, curve of open circles, reaches a maximum at a plasma concentration of about 5 mg%, but is constant beyond that. Obviously, as noted before, there is a maximum capacity of the tubular cells to perform active transport, or apparently there are intracellular carriers capable of shifting a limited load. The structural and physicochemical nature of such carrier systems will be discussed later. Some comment is still necessary concerning the ascending part of the curve, picturing the tubular transfer. Evidently, this part signifies that, starting with P equal to 0, the efficiency of the transferring system becomes less and less until the maximum uptake has been reached at 5 mg% in the experiment question. In other words, the accumulation ratio falls off with rising concentration, as has been noted in various earlier observations. It is particularly obvious with active systems, where passive penetration by filtration is negligible, for instance. In the case of the intestinal absorption of glucose, P544, or with certain plant correlating the results of experiments on various animals, the ratio between active transfer and passive filtration has been found to differ systematically from species to species. In other words, the tubular function can take precedence over, or be subordinate to, the glomerular function. The latter may even be negligible, as in the aglomerular fish. As a matter of fact, when phenol red is injected into the goosefish or the toadfish, P568, the effect of tubular excitation causes the dye concentration in the urine to rise with increasing plasma concentration to a maximum in a manner similar to that shown in figure 6 by the curve with open circles. But there is nothing in the picture of the total excretion by the aglomerular kidney that can be compared to the ascending curve with dark circles due to the additional glomerular filtration. Consequently, it is highly significant to the behavior of the aglomerular kidney that the phenol red concentration in the plasma can exceed considerably the maximum concentration in the urine, indicating that, except through active transfer, the tubular wall is inaccessible to the dye. So far, the report about the influence of secretion and reabsorption upon the plasma clearance has been chiefly confined to experiments with phenol red, which from earlier experiments with other methods was already known to be pushed through the tubular wall in the direction blood illumine. Clearance determinations with some other substances likewise have led to conclusions which more or less coincide with the previous results. It has been mentioned before that the differences of the clearance values before and after fluorescent poisoning indicate that glucose and xylose are reabsorbed unaffected by the presence of the poison. The same conclusion was drawn from frog perfusion experiments, P561. Further, according to both methods applied to frog P562, and dog amino acids seem to be reabsorbed studies upon frog, nectarus, and dogfish, namely active secretion, indifferent set, and active arc-absorptee, P572. The clearance method applied to chicken, dog, and mben has given evidence of possible reabsorption. But this may be better described as a passive escape through diffusion because of the low molecular volume of urea. According to the formerly reported results with the frog and the lizard P573, uric acid undergoes active secretion, but clearance determinations in Manare interpreted by Smith as indicating active arc absorption. This section of the book, dealing with passive penetration and active transfer in animal and plant tissues, has opened with the analysis of intestinal absorption and of urine formation, because this analysis brings up a large number of subjects that have to be discussed repeatedly in the following chapters of the section. In the performance of their function, both of these organs, intestinal mucosa and kidney, must dispose of many diverse substances, mixtures of foodstuffs in the one case, mixtures of blood components in the other. Both organs are compelled to admit some substances according to certain general physicochemical properties. But in addition, in the intestine, special provision is made for the intake of substances, which are necessary for the satisfaction of special metabolic wants. And in the kidney, there are other arrangements empowering the output of other substances either useless or detrimental to the body. Obviously, it is at least partly due to these selective activities that the rather definite concentration levels of the blood fluid, so essential to the constant life of the higher animal, are secured. And it is an important problem. As mentioned once before, pages 554-571. To test whether these teleologically conceivable selective capacities can be correlated to certain chemically or physicochemically understandable affinities between the dissolved substances and the constituents of the epithelial membranes. It is the plan to carry on the functional analysis of other transporting organs, which are more specialized in their physiological accomplishments, and correspondingly display an ability to select different substances for an active transfer, which is more restricted than that of the intestine and the kidney in their function of checking the passing substances at one of the main inlets and one of the main outlets of the body. According to this plan, the following organs will be studied. 1. The body surfaces of animals and the root system of plants. The discussion of their properties will raise the question of osmoregulation to it. The digestive glands, the salivary and the stomach glands, pancreas and liver. 3. The membranes separating the blood fluid and the aqueous humor and the cerebrospinal fluid. It is obvious that, in many regards, this discussion will be limited to those living objects which normally are in intimate contact with an aqueous environment. The problem of natural permeability of their surface seems to be simplified by the fact that, in general, the permeability to non-electrolytes can be neglected compared to the permeability to water and to inorganic ions. The main information regarding animals cannot be acquired in relation to its aqueous environment. But then it must be remembered that the permeation of water and electrolytes through the surface may be accompanied by the simultaneous intake and output through the intestinal tract, the kidney, the gills, and other organs. The amphibia alone provide the opportunity to investigate the properties of the isolated, undamaged surviving surface, since in them the skin is only loosely attached to the body, and therefore rather uninfluenced by the processes going on in the interior of the body. Concerning the properties of plants, the studies will be restricted here to the roots, including some functionally comparable systems. The osmotic properties of the marine invertebrates. For a long time, the more general concepts about the skin permeability of animals living in an aqueous surrounding were derived by Batazzi, Frederick, Duval, and others from comparing the osmotic properties of the internal environment and the external environment, milieu interne and milieu externe of Claude Bernard. It was stated that, under ordinary conditions of life, the two media are approximately isotonic in the case of the marine invertebrates and the marine elasmobrates. In other words, these animals are in osmotic equilibrium with the seawater, that is, at equals O2 whereas the body fluid of the marine teleos is hypotonic, at equals about 0.8 to 1,1, and that of the freshwater invertebrates at equals about 0.1 to 0.8. The freshwater teleosts, AI equals about 0.550, and the freshwater amphibia, This suggests the belief that maintaining a difference of osmotic pressure of several atmospheres across a thin cell layer, as affected by the marine teleosts and by freshwater invertebrates, teleosts, and amphibia, requires regulatory arrangements which may be unnecessary in the other groups. In order to test this hypothesis, observations upon the osmotic properties of G. fongiae marine invertebrates may be quoted first. Here the Mediterranean Sea AO 2.8 degrees of the body fluid of Mytilus agelou was found to be 2.1 degrees, while in seawater of the Baltic Sea of a certain provenience equals 0.94 degrees. It was 0.95 degrees. Similarly, in the North Sea, AL equals 1.7 degrees. AA of Ornithola marina was 1.7 degrees. In water of the Baltic Sea of another provenience, ACO equals 0.8 degrees. 0.75 degrees. These osmotic equilibria in animals living in far distant habitats can be explained as simply being reached either by the passive exchange of solutes or of water, or possibly they are due to complicated adjustments over a long period of time. However, when in an acute experiment, a Plygia, a marine snail, suddenly is transferred into 75% seawater, the weight increases within 1 hour about 25% and falls during the following 6 hours below the original weight, and the body approaches a lower osmotic equilibrium dace, as will be shown later. This is probably due to nothing more than, as in a common osmometer experiment, an initial rapid osmotic uptake of water, followed by a slower escape of solutes. But there are other observations on marine invertebrates suggestive of the existence of special osmoregulatory process. ECSL, when Carcinus maenas at equals 1.75 degrees is transferred from seawater or equals 1.4 degrees to diluted seawater L equals 0.68 degrees. It drops down to 1.8 degrees within 26 hours. But thereafter, this difference AIAO 1.8 degrees, 0.68 degrees remains constant throughout the subsequent 15 days. It cannot be inferred that this is caused by the slowness of diffusion across the exoskeleton of the exoskeleton crustaceans, because a similar reaction is observed with a marine annelid, Nereis diversicolor. Presumably, those invertebrates which, living in brackish water, are subject to frequent and large concentration changes of the external environment, are especially able to compensate in some way for the fluctuations of the osmotic. The belief that the aforementioned body weight or volume changes of a Plygia are attributable to penetration of both water and solutes is strongly supported by the experiments of Beatty, demonstrating particularly the passive permeability of the body surface to inorganic and organic compounds, which appears to be much more widespread than was earlier assumed. After a Plygia has been transferred into 3 parts of seawater plus 1 part of isotonic sucrose, the weight does not remain constant, as might be expected, but diminishes progressively, CG, within 20 hours to about 50% of its initial value. The reason is that after the animal has been immersed in mixture, the salt concentration is lower outside than inside, hence salt leaves by diffusion. As the result of that, the osmotic pressure becomes lower inside than outside and water escapes. When, in an experiment of this kind, sucrose is replaced by glucose or glycerol, the decrease of weight is smaller with glucose and still less with glycerol, as glucose passes more easily into the animal than sucrose, and glycerol still more rapidly. More direct evidence of inorganic ions being able to penetrate in one or the other direction has been afforded by placing C7RC yin alternately in artificial seawater, either containing the main cations and anions in normal concentration, or having a deficiency or a surplus of one of them. For example, Kaji, one of the results is the following. Artificial seawater. Artificial seawater. It is obvious that in this experiment only shows a decrease of concentration in the blood corresponding to the outside deficit. The loss of blood often is accompanied by a loss of tonus and of reflex activity. A similar shift of ion concentration was obtained with Ka-Mg and Ki-L. It may be objected that in experiments like these, the exchange of ions does not take place across the hard shell, but rather within the intestinal tract. However, the same result is obtained after closing the mouth and the anus. Direct proof of penetration of the shell has been furnished by cementing a metal cylinder upon the dorsal side of the carapace and filling it with dilute milk. The iodide ion, which appears in the blood, is believed to pass chiefly across the small holes of the shells, which are occupied by cutaneous sensory organs. Penetration of ions across the body surface has been shown to exist, but even seemingly to predominate over the water exchange, as followed from the slight changes of body weight under anisotonic conditions. However, this test is fallacious. In reality, for example, with the animal in dilute seawater, the water can enter the body by osmosis, but only in an amount equal to that of the fluid lost through ejection of jets of gastric juice, urine, and secretion of the antennal glands accompanying the increase of hydrostatic pressure inside the exoskeleton. This is one manner of osmoregulation under the special anatomical conditions of hard-shelled animals. So far, one might be led to conclude, from the majority of experimental data referred to, that the body surface of the marine invertebrates is really accessible to the components of their external media, like a sponge. But this concept is inconsistent with the fact that the percentage of individual inorganic ions found in the blood serum of these animals under natural conditions seems to diverge considerably from their percentage in the environment, and to display species-specific differences. Thus, with certain crustaceans, the Mg percentage is remarkably lower, the K percentage higher, than in the environment. Albon, this would be incompatible with a merely physicochemical viewpoint of passive diffusion and osmosis, but could be attributable to physiological regulations, which, under the artificial conditions of an experiment, could be overshadowed by unusually gross effects. In any case, this conflict possibly will be abated by observations referred to in the following 2 paragraphs, which have furnished clear evidence that under fairly normal conditions, a slow active transfer of very slight amounts of ions across the body surface can be effectuated by teleosts and by amphibia. 2. The osmotic properties of marine and freshwater teleosts. On the basis of present knowledge, it is believed that a steep and persistent concentration gradient between outside and inside is significant of the majority of teleosts. Approximate values of the gradient for marine and freshwater teleosts are listed in the following table in terms of differences of freezing point depression, of atmospheres, of osmotic pressure, and of molarities. In general, both classes of fish die a short time after the normal concentration gradient has been changed, with the marine animals by diluting the seawater, with the freshwater animals by adding it. But there are some observations showing that if this change of the surroundings is accomplished slowly enough by the gradual addition of more and more water, it was mentioned previously, p. 581, that the marine osmobranch resemble the marine invertebrates in that both groups of animals are in approximate osmotic equilibrium with their surroundings. But they differ in that with elasmobranch, no more than 40 to 50% of the osmotic pressure of the blood fluid is due to salts. The rest is chiefly due to urea, P572. In this respect, the blood of the elasmobranch is comparable to that of the marine teleosts, which is hypotonic to seawater, P81. Concerning the interesting complex osmoregulatory faculties of this class of animals, and the particular role of urea, see C. Schleiper, by all Reverend 5, 31930, H. W. Smith, M.J. Hiziel, 98, 279, 296, 1981, or salts respectively, death can be avoided. This is shown, for instance, by the following experiment upon Saprinus carpio, KGT xipsa. Adjustment of a freshwater teleost Saprinus to saltwater experiment duration. Days it appears that with stepwise increase in outside concentration, the inside concentration rises and retains a higher level over a long period. Obviously, the slow change has stimulated some adjustments, though not an osmoregulation. Possibly light may be thrown upon the problems of such a gradual adjustment by the following observations upon the marine teleos, Opsonus tau, toadfish, and Myosex Dutch phallus dies. The flow of urine is very slow, and the urine is free from chloride. The plasma chloride is not higher than 150 millimole. But even a slight injury of the skin, the avoidance of which requires extreme precaution, is followed by an enormous rise of urinary flow, and of urine chloride 200 millimole and more, and by a marked increase of plasma chloride, possibly accompanied by increased swallowing of seawater. It seems not unlikely that the aforementioned adjustment of saprinus is due to avoiding sudden changes of the osmotic conditions on the body surface, which would be succeeded by damage and disruption of the epithelial layers. Most distinct osmoregulatory capacities are observed among the teleos with those species, which, due either to migratory life, eel, salmon, or to their living in brackish water, are accustomed to great changes in the eye, it appears first. That after the first day of each period, there is only a slight change of the internal osmotic pressure, and second, irrespective of the hypertonicity or hypotonicity of the external media. The internal media are kept fairly constant, but this result appears only in case the surface of the body remains intact. It does not occur when the surface is exposed to an unbalanced electrolyte solution, Sex 4 and 5, instead of seawater, for example. When seawater is substituted by an isotonic nasal solution, the fish becomes very weak, and of its body juices increases from 0.89 degrees to 1.1 degrees after only 1.5 hours, indicating an abnormal increase of permeability of the body's surface. Information concerning the location of the osmoregulatory mechanism has been provided by experiments upon the eel. For various reasons, the gills were looked upon as being of prime importance in regulatory activity. In an effort to test this hypothesis, the following procedure was applied. The head of the eel was severed from the body, and a heart-gill preparation established with the heart pumping in a closed system, a constant amount of the internal medium through the gills and returning it through the dorsal aorta to the auricle, while in another closed circuit, the external medium, coming from a reservoir, entered the oral cavity and, leaving it by way of the branchial apertures, returned to the reservoir. A Ringer-Lock solution of suitable concentration was used as internal medium, taking into account that for freshwater eels, A of the blood was reported to range from 0.56 degrees to 0.63 degrees. For seawater eels, from 0.64 degrees to 0.74 degrees. The result of analyses of the two media, which control each other, leaves no doubt that, in seawater eels, chloride is shifted against the concentration gradient from the perfusion fluid through the gill membrane into the seawater, which contains Cl in about 3 times higher concentration. This shift seems to increase with increasing Cl concentration in the perfusion fluid. With fresh water as the external medium, the gills seem to be inactive. They allow water to enter the perfusion fluid, and a surplus of water is believed to be eliminated by the activity of the kidneys, which, under these conditions, excrete a large amount of very dilute urine. Reasoning that the function of active transfer of chloride ions might be reserved to specific structures in the gills, which by a histological investigation could be differentiated from the respiratory elements, just as in the stomach glands. The parietal and the non-parietal cells have been distinguished as HEL and as enzyme-producing units. 599, Keyes and Wilmer. The heart-gill preparation also has served to expand previous observations that a considerable excretion of NCOMs, especially of urea and ammonia, takes place in the gills. H.W. Smith again has fixed fresh water teleos in a 2-compartment tank in such a way that the water surrounding the head and the body can be analyzed separately. JTI pairs that the branchial output exceeds the urinary excretogy by several fold. There is no reason in these experiments not to ascribe the urea output to a simple diffusion. since the concentration of the blood urea was always found to be higher than that in the water around the gills. With the heart-gill proper ration, considerable amounts of urea can be detected in the external medium only when the internal medium contains Jureti Ij3, the frog properties of amphibia, the frog. Living in fresh water, aka 0.1 degrees, maintains its internal medium at a level of H.43 after having been kept dry for a while, resulting in a loss of 25 to 30% body weight. The frog regains the lost weight in moist surroundings even when water uptake by mouth is prevented. In water, the weight increases progressively after the cloaca has been closed because the abundantly excreted urine accumulates in the intestinal tract. If, under such conditions, the frog has plaque dye in salt solution instead of water, the increase of body weight diminishes with increasing salt concentration and changes to a decrease at about 0.8% Nakelta. These observations of Overton are indicative of a skin permeability to water in either direction. There is further a definite, though low, permeability to dissolved substances, particularly to inorganic ions. This is shown by the fact that after placing the frog in KCl solution, K appears in the urine in considerable amounts, but no symptoms of K intoxication become evident even in isotonic KLL, because the K level in the blood is kept low by the activity of the kidneys. According to Crow, frogs living in fresh water suffer continuously a small loss of salt through the excretion of large amounts of urine. If no food is taken, and if the surrounding water is renewed frequently, or the frog sprayed for a long period with distilled water, the stores of body salts are depleted and the animal grows weak. In this state, the skin displays a marked absorptive power from 30 to 50 cc of 3Q. Ringer's 6.5 milligrams. Percent nickel, KLL is completely absorbed within a few hours. This absorption is not due to an osmotic inward current of water, for it also takes place through studies of the microscopic picture of the gill epithelia of teleos, including the eel have discovered on the bases of the gill leaflets. Special cells far exceeding in size the predominant respiratory epithelia, and have discussed the justification for calling them chloride-secreting cells, A. B. Keyes and Eden Wilmer, J. Physiol, 76, 368, 1932, in isotonic sugar solution from which no water is taken up. Bar is absorbed at the same rate as Cl, but I is not absorbed together with Abi. This shows the absorption to be an active transfer by the skin. K together with Cl enters only in small amounts compared with Na plus Cl. Kia does not enter or doshin, so only slightly. The frog skin offers a rare opportunity to investigate osmoregulatory power, since, because of its loose attachment to the body, fairly normal pieces can be cut out as diaphragms for separating two fluids. In 1892, W. Ryden's degrees began this kind of experiment with the intestinal mucosa, P5 and 52, which repeatedly failed to give definite results because of the great frailness of the mammalian tissue. But he was successful with fresh frog skin in showing that this membrane, fixed as a diaphragm in a differential osmum tier, the two compartments of which were filled with Ringer solutions, establishes a hydrostatic pressure difference by shifting the solution from outside to inside. This transport also takes place against a hydrostatic pressure of 2 to 4 centimeters, water established at the beginning of the experiment. But after the skin has been treated with M-luoxen, the transporting power is abolished. And if a marked pressure difference was already established, the application of XNCN results in a reversed movement of the fluid until the difference has been abolished. These various observations provide clear evidence that the experiment of Reid is a demonstration of an active transport of water. However, beyond this, there is an active transport of solutes, as in Crow's experiments on the whole animal. This has been shown as follows. The skin of the two legs is stripped off and the two cylindric bags are used like vessels of an osmometer. They are filled with measured amounts of Ringer and are dipped in Ringer. In different experiments, the skin sacs are oriented differently. Either they are used as they appear after stripping inside out, or they are used in their natural orientation. According to the aforementioned observations, these osmometers, turned either way, can be expected to shift fluid from the exterior to the interior surface of the skin, the two sacs controlling each other. However, when one sac of a pair is exposed from both sides to a poison, not only is the shift of fluid diminished or stopped, but as shown in Table 51, the percentage of Cl in the ringer solution is changed also. It appears that in each of the 5 experiments, the concentration of Cl is diminished on the natural outside of the skin. In other words, Cl is shifted from outside to inside, yielding a higher concentration than that initially present on both sides. And this seems to be dependent upon a supply of metabolic energy, as can be concluded from the effect of poison. KCN blocks the oxygen consumption of the skin bromoacetic acid, which is similar to iodoacetic acid, the production of lactic acid, which normally is subjected to aerobic breakdown. Therefore, see experiment 5. The concentrating ability of the skin after having been depressed by bromoacetic acid is improved by the addition of lactate. These results will be discussed more in detail at the end of this section, pages 97f. In conclusion, it may be added that the accumulation of Cl at the inside of the skin disappears as the skin dies. Active transfer of Cl from outside the skin to inside increase or decrease of equals 1 contained inside the saki dura bromoacetic acid dura TNHS5. After bralatate, the problem concerning the location of the activity will be referred to for irreciprocal permeability of the frog skin. Before the active transfer of dissolved substances directed across the frog skin from outside to inside was discovered, a preferential unidirectional passive movement of solutes, as well as of solvents, had been observed and analyzed in many studies. This irreciprocal permeability is due to various factors which together result in some kind of asymmetrical composition of the skin that, in the first place, is structural in nature, i.e. the skin is built up by a number of strata. See also P 319. These are 1. Different in their chemical composition, and for this reason can react specifically with the penetrating substances, especially do they differ in pH 2. They are solvents of greater or lesser dissolving power toward the substances. This will result in the formation inside the skin of diffusion gradients of different steepness and even of different direction. 3. Their mechanical resistance to the entrance of various components of the solution differs and can vary insofar as, for example, the same electrolyte solution can alter only the inside or the outside of the skin. 4. They are built up by colloids of various nature which, e.g., under the influence of the same ions, will either swell or shrink. All these factors can influence each other in such a way that diffusion or osmosis across the skin is directed either inside-outside or outside-inside. 5. Active transfer in some plant tissues. The general picture representing the fundamental properties of the plasma membrane in allowing passive entrance of organic compounds into the cells has been outlined mainly on the basis of experimental studies of plant tissues by 2 botanists, Overton and Collender. G also, chap 10. In this picture, the G-plasma membrane is shown to act partly as an organic solvent and partly as a molecular sieve. However, it was also recognized very early that upon such a stable framework there must be superimposed dynamic devices able to bring about the complex mixture of substances making up the intracellular contents. This was most obvious in studies of inorganic ions, penetration of which cannot be accounted for without assuming that the concentration gradients varying in steepness as well as in direction, P244, are the result of driving forces regardless of the very common experience that inorganic ions, when studied by osmotic, chemical, or electrical methods, do not display, or hardly display, an ability to pass the surface of the plant cells. But during the last 2 decades, it became more and more clear that this BC-Weldering discrepancy, which is most obvious in plant cells, rests upon the 2 facts. First, That indeed, the rate of diffusion of ions in either direction across the protoplasmic wall is practically zero. And second, that independent processes, unlike diffusion, establishing an unbalance, also go on with great slowness, and even at all, only provided a sufficient supply of energy can be furnished by the cell in order to start the specific machineries in one or the other direction. In the earlier permeability studies, measurements were made mostly by observing rates of plasmolysis or deplasmolysis with single cells, or with a small number of cells by observing conductivities, or applying chemical and spectroscopic methods in experiments frequently too short in duration and dealing with too limited amounts of material to allow the detection of small changes in concentration and composition. Great progress in this field is due to F.C. Stewart and W.R. Hoagland, who have developed methods for the utilization of large quantities of plant tissue, which, if properly prepared, gives a homogeneous material suitable for observation over a considerable period. E. Overton, Vierteljahrschrift, Naturforschung. Chess in Zurich, 41, 1895, 44, 68, 1899, R. Collender, and H. Berland, Actabat Fen, 11, 1933. Of young roots or storage organs are placed in weak solutions of certain salts, which can serve as an adequate medium. Since under normal living conditions in the soil, these organs also are in contact with such solutions in small amounts, serving as a source for their ionic needs. Insofar, they may be compared to the freshwater amphibia and Telosti described methods. Storage organs which have been found useful are carrots, artichokes, turnips, dahlias, and especially potatoes. A great number of discs of these organs, 1 to 0.75 millimeters, in thickness, are placed in water and are well aerated for several days. By this treatment, the dormant starch-filled cells become more and more depleted of starch. Their slow respiration rises, protoplasma streaming becomes visible, and, accompanied by the revived aerobic metabolism, salts, which in low concentration are added to the water, pass across the thin protoplasmic wall and are accumulated in the cell sappy. Roots have been used in the following way. Why seedlings of barley are grown in diluted nutrient solution. Thereafter, in order to avoid complications arising from interrelation between root and shoot, the roots are excised and are kept well aerated for about 10 hours in the experimental salt solution. The result of these treatments can be determined by freezing and thawing the lots of cither discs or roots, pressing them and analyzing the fluid for mineral content. This procedure has been proved to yield fairly, since it is rather generally agreed that from equivalent solutions, the cations mostly arc, absorbed at a rate decreasing. In the series Kens so far, M and G, the anions in the series Na3 Most experiments regarding absorption have been performed with Na3 and KCl. In many experiments, it has been found useful to substitute for Cl, the near-related Br, eventually also Rb for K, in order to secure conclusions by employing non-toxic ions, not initially present in the cell's results. Through experiments of this kind, it has been found that, starting with very dilute solutions, cations and anions enter the cell with rising concentration against their gradients, but in such a way that the accumulation ratio is higher. When the tissue is exposed to lower concentrity ions, this is shown in the following table concerning the absorption of KBr by carrot discs from solutions varying 100 times in strength. Concerning studies on single cells, particularly on giant cells like Valonia nidla cara, with respect to active transfer and passive penetration of inorganic ions, as described by Osterhout, S.E. Brooks, Collender, and others, see Chap 11 Final Internal Cone. This accumulation cannot be referred to binding forces, since the analytical results and the measurements of electrical conductivity of the sap are in fairly good agreement. The maximum value in this experiment is relatively small. With barley roots exposed to very dilute solutions, accumulation ratios, which are even in excess of 1,0, have been reached. The lower values observed with discs of storage tissue are dependent upon the thickness of the slices as well as upon other factors. The thinner the discs, the greater is the accumulation. During the aforementioned long preliminary aeration of the discs, according to microscopic evidence, only the outermost layers of the initially dormant cells are reactivated. That is, for potato, a layer of about 3 cells, equal to 0.35 millimeters thickness, whereas the discs already narrowly employed were about 0.75 millimeters and more in thickness. The vitality of the accumulating power can be proved in many ways. First, by taking advantage of the fact that it is intimately connected with the metabolic activity, or better, with the aerobic respiration. This is illustrated by the following table: 6 additional hours in air. It appears that the transfer of K in bar, which is high in air, falls off markedly in N2, but is restored in a following period of air treatment. The table further shows that cog production, which is a regular indicator of aerobic respiration of an active organ, fails to display a deep decline in N2, proving that the active transfer needs O2, and that anaerobic CO2 production apparently is not involved in this function. Correspondingly, MB500 to M0 KCN, which depresses O2 consumption more than CO2 production, is an effective and reversible inhibitor of the uptake of K, but even the retention of salt, which has been already accumulated, is ensured only in the presence of phenylate. It is particularly interesting that anaerobically and to very little K and BRAR enter the roots, even though a marked inward gradient exists for these ions, whereas aerobically both of them are not only absorbed, but even accumulated to a higher concentration than that of the relatively strong external concentration. Evidence from many experiments has been obtained that the accumulating process usually has a more complicated aspect than so far described. In the plant tissues, Steward USC has differentiated I-Kytween primary absorption and induced absorption. The first is defined as an uptake where cation and anion simultaneously, in equivalent amounts, enter the cell sap, whereas in induced absorption, external and internal media influence each other by an exchange of their different components, the net effect being an increase of the salt content. This exchange appears in various forms. For example, from a solution of K2-VO4, a greater amount of K is removed than so far, with the result that the external solution becomes acid by an exchange of internal H for K, while from a solution of T-3, 2 more, no enters the cell than Ti. And this causes alkalinity by the escape of HCO3. The amount of such exchange depends on various circumstances, for example, on the metabolic activity determining the magnitude of active transfer, or on the concentration of exchangeable ions accumulated during the period of growth, high salt and low salt roots, or on the existing about the differentiation between accumulation of Rb in the protoplasmic mantle and in the cell sap of Vauxnia and Nutella. See the studies of S.C. Brooks, Sec 5, P345. 7H, accounting for the metabolic production of exchangeable Uday organic acids and other factors. However, these various factors, which obscure more or less the clear effect of primary absorption, HUIR, a great significance, mainly LLNE, at accumulating power of the tissues has subsided, or has not fully developed, whereas the primary absorption unmixed with the passive exchange is most conspicuous in vigorously aspiring and growing systems. Examples of such active transfer of cation and anion in equivalent amounts have been obtained with various objects. An example is shown in figure 63. The uptake of the ions was followed through a period of about 100 hours. It is evident that in the J area, 21 hours to 96 hours, the absorption of anions and cations is in approximately equal amounts. During the first 21 hours before samples for the analysis are collected, by some manner of induced absorption Kb must have entered with greater rapidity, while for Bror there is a period of lag of very slow absorption. In conclusion, it should be emphasized that the essential features of the active transfer in plant tissues strikingly resemble active transfer in animal tissues. As to the transfer of salts, the simultaneous absorption of cation, Na and anion KL, by the kidney epithelium, although not strictly proven, can hardly be doubted. since nickel, after being filtered through the glomerular membrane into the Bowman capsule, disappears completely during passage down the tubules P559. The same type of active absorption of Na and Cl ions is effective in the frog skin P508, whereas in the intestine, this kind of shift appears dimmed due to the greater importance of passive diffusion in the penetration of this membrane. Another point of similarity regards the inverse relationship between the outside concentration and the accumulation ratio P592. This relationship was found very conspicuous in the kidney in the secretion of urea and of diastuffs P573, and will be found in the secretion of diastuffs by the liver P600. This has led to the assumption of carriers being involved in the transport into and across the cells, and being differentiated to accept only specific loads, and these only up to a maximum amount. Whether such a hypothesis will suit also the conditions observed in plant tissues awaits further investigation. As lias been enunciated before, focusing in this section our chief interest upon active transfer across layers of living cells. We have seen that intestinal and kidney membranes are most versatile in performing this kind of work, in contrast to the body surface HF animals and plants, which appears to be much more specialized in transporting ability to inorganic ions. The latter behavior is likewise characteristic of the digestive glands. Defining digestion as a process which performs or supports the chemical breakdown of foodstuffs, the digestive glands comprise mainly salivary glands, stomach mucosa, pancreas, and intestinal mucosa. The chemical breakdown is brought about in the first place by specified intracellular products, the enzymes, which in general appear to be included in granules. The discharge of these contents is dependent upon innervation. But very little knowledge about this accomplishment has been made available by physiological study. What is known is mostly morphological in nature, such as details of the microscopic structure of the secretory cells, displayed by the living as well as by the preserved materials. However, the process of the formation of the digestive juices, that is, mainly saliva, gastric juice, pancreatic juice, and the composition of these fluids as compared to their source, the blood plasma, is accessible to physiological experiment, which is chiefly concerned with studying the ultrafiltrate of the juices, that is, neglecting the colloidal substances, which are mainly the enzymes, but stressing the elucidation of the forces involved in the segregation of the fluids. There is only one more glandular secretion participating in the digestive processes, the bile, which stands apart first because of its lack of enzymes. Second, because of the content of special characteristic organic compounds in great amounts, namely the bile salts and bile pigments. Third, because its discharge is independent of innervation. 1. The formation of the salivary fluid, disregarding the colloidal components of the saliva, enzymes, proteins, mucin. The salivary fluid is mainly a very dilute solution of inorganic salts, to which, in relation to the degree of activity, varying small amounts of lactic acid, phosphate, and other substances are added as split products of glycogen and phosphocreatine. Ordinarily, saliva is decidedly hypotonic to the blood plasma, and accordingly chemical energy is spent in order to perform the necessary osmotic work. The freezing point is found to vary in a wide range, and is correlated to the species, to the kind of gland, submaxillary parotid, to the height of glandular activity, and to the kind of innervation, sympathetic, parasympathetic. Frequently, at submaxillary saliva is found as low as 0.1 degrees to the ionic composition of the salivary fluid is different from that of the blood serum. Most striking is the surplus of K in the saliva, and the ash of saliva shows an exceedingly high percentage of K. It may be asked whether the ionic composition is the result of some kind of selective permeation. Another possibility would be that the source of the KCr+ is the protoplasma of the cells, which, discharging their granular contents through their inner surfaces into the lumens, could lose some K, referring to the first alternative. The permeability of the salivary gland has been studied in different ways, either in experiments on the total animal, rabbits, or on the isolated submaxillary gland, cat. artificially perfused with hemoglobin ringer. Various substances were added to the circulating fluid, and the saliva produced after electric stimulation of the chorda was analyzed. The results obtained bear upon 1. Organic non-electrolytes selected with regard either to their molecular cyste or to their lipoid solubility. 2. Dyestuffs. 3. Inorganic electrolytes. A. Organic non-electrolytes. Among these non-electrolytes, neither disaccharides UMV equals 346, nor hexosauce, glucose, galactose, fructose are apt to enter the saliva, but the trioxide dioxyastin does, Tastro 70. Alanine 250 FF-glycine 110 EFF2A, and melanamide either do not penetrate or only slightly, while propionamide and particularly acetamide pass with ease. When melanamide is calm, paired with butyramide and dimethylurea, the molecular volumes of these three compounds, being about the same, butyramide and dimethylurea, are found to enter the saliva in a considerably greater amount than melanamide. This is very likely to be due to the influence of their lipoid solubility, obliterating the influence of the molecular volume. One irregularity has been observed in the perfusion experiments, which so far cannot be accounted for. Urea passes markedly more slowly than acetamide, although they resemble each other in their small molecular volume and in their lipoid insolubility. It may be recalled that also in other regards experiments with urea turned out to give unexpected results, p. 572. D. Dyestuffs. 3 acidic dyestuffs, which have been tested, indigocarmine, fluorescein, and erythrosine, do not pass into the saliva. C. Inorganic ions. The behavior of the inorganic ions is particularly pertinent to this subject. With regard to anions, it has been stated that after intravenous injection, sulfate, phosphate, and ferrocyanide failed to enter the saliva further. The lower values of 0.1 degrees and 0.25 degrees are enough proof that, at least under certain conditions, CP 598, KL does not pass readily. Only iodide was found in the parotid saliva after an intravenous injection of 20 milligrams, nal per kilo. It appeared 7 times concentrated. Regarding the cations, there are data shown indicating that at rest K and Na are predominant over Ki and Ng. This order is corresponding to what has been observed with the intestine, pages 586, with the frog skin, p. 588, and with plant tissues, and taking into consideration that sugars and amino acids do not pass, and that saliva in general is markedly hypotonic to serum. One may say that the salivary gland often behaves as though very little permeable to solutes. However, it is important to notice that this picture changes somewhat when the glands are thrown into greater activity by any kind of stimulation. According to the old observations of Heidenhain, the salt content of the saliva is raised by raising that even may approach isotonicity. For instance, the nasal percentage in the chorda saliva of the submaxillary gland, dog, as calculated from the CI values, can increase from 0.92% to 0.55%, with 0.7% in the serum. And these results are confident by figures for Na and 1 present blood in it. Appears first that during rest there is slightly more JV in the saliva than Na, but that during activity it prevails by far. Second, that during activity Na is increased 8 times, whereas X remains unchanged. This suggests that the output of Na salt and the output of K salt are somehow independent processes. Perhaps this can be interpreted as an increase of permeability of the activated gland cells in such a way that NACL, which is present in the serum in a paramount concentration, and is preponed in the serum even during great activity, see table 54, enters T cells together with water across their outer surface and passes into the secretion, while K may continuously leave the cells through their inner surface as wear and tear product of the protoplasm. In order to test a concept like this, the reappearance in the saliva of Libcha and Einschagel, of SO4 and IPO4, presented in varying concentrations to the resting and to the active gland from the circulating fluid, should be studied at 27. It has been mentioned before p. 595 that during stimulation, lactic acid and inorganic phosphate appear in great amount in the blood and eventually in the saliva as split products of glycogen and phosphocreatine. After poisoning the isolated hemoglobin linger perfused submaxillary gland with cyanide, the discharge of lactic acid and iphosphate into the perfusion fluid is increased. After iodoacetate, that of lactic acid decreased. Of phosphate increased. These effects of poisoning are associated with concentration changes of the ionic output into the saliva. The release of Cl is reversibly raised, and under these conditions, X likewise is set free reversibly and in a great amount, even so much so that it seems to surpass the concentration in the perfusion fluid. This would be indicative of a heavy, though reversible, loss of intracellular X from the above. The following fragmentary picture of the process of the secretion of salivary fluid may be drawn. The essential work to be done is the segregation of water from the blood fluid, serving the purpose of wetting. 227, according to Höber and Stavriky, Court J. Exper, Physiol, 26, 141, 1936, the supraxillary gland, which during stimulation of the chorda, or the sympathetic nerves, remains glucose-tight, P. 596, becomes permeable to glucose following administration of adrenaline, when the sugar is injected intravenously. During hypoglycemia, produced by starvation and florazin, a relatively high dose of adrenaline, in addition to nerve stimulation, throws into the saliva a small amount of sugar, which immediately and markedly increases in amount when glucose is injected, imbibing and swelling of the food materials. This segregation is initiated breakdown of organic substances. The coincident movement of inorganic ions is a mere accessory phenomenon due to the incomplete semipermeability of the outer surface of the active cells, which allows the Na and Cl ions to take the steep downhill path from the plasma through the cells into the secretion, and perhaps due to the permanent leakiness of their inner surface decay. The organic metabolites leaving the cell evidently are diverted more toward the blood fluid than toward the saliva. It is unknown how this is accomplished. 2. The formation of the gastric secretion. As in the previous section, and for the same reason, in the following outline of gastric secretion, all those observations will be omitted, which mainly on the basis of biochemistry as such, and from histological evidence, have contributed to the present knowledge concerning the action of the gastric gland in the elaboration and the elimination of enzymes and of mucus. In other words, interest will again be centered around the composition and the forces which are involved in the production of the abundant aqueous ultrafiltrate like gastric fluid. The problem concerned is very different from that encountered in the study of the salivary secretion, since the gastric secretion is nearly isotonic with the blood fluid, and therefore needs no energy supply for the shift of water. And since by its composition, undoubtedly, active transfer of solutes is manifested as demonstrated by the fact that, due to its main constituent, 2-ECL, the secretion is about 3 million times more acid than the blood fluid from which it derives. For several reasons, it has been assumed for a long time that the parietal cells of the gastric glands are the seat of HCl production. But the parietal cells are not more than a small fraction of all the cells of the gastric glands. Non-parietal cells, which include the so-called chief cells in the surface epithelia, are supposed to elaborate the pepsin and the mucus. However, aside from these colloidal substances, gastric juice, besides secon and TL, also contains other ions, Kna, Kmg. Therefore, the question may be raised whether the gastric fluid is not the result of the simultaneous activity of several glands. This question has been attacked mainly by Gray and his co-workers in the following way. A continuous secretion of gastric juice is maintained by subcutaneous injection of histamine at 10-minute intervals, and the collected fluid analyzed for the concentration of HK and NaCl with reference to the volume rates of secretion. The results are listed in the following table. It appears that the concentrations of Cl in millisecond liter surpass those of H, that with rising volume rates CCl 20 min. H, expressed as ECl, increases much more than does TL. and that the concentration of H at the highest rates of secretion approximately reaches the concentration of Cl. This is evidence of an independent secretion of HCl and a neutral chloride BCl. The concentrations of neutral chloride are calculated in the table as the differences of the concentration of Cl and HGl. This shows that the fraction of neutral chloride secreted falls off steeply with rising concentration of total chloride, so that the output of neutral chloride finally becomes rather insignificant in comparison to the total output of HCl. This is especially evident from the extrapolated results, in brackets, indicating that at maximum volume rates of secretion, the concentration of the gastric fluid is equal to 154 milliecu HCl plus 12 millicuv Cl. Probably the volume of BCl secretion is small and increases very little. Compared to the large increase in the output of HCl from the table, it furthermore appears that the concentration of BCl has been fractionated into those of the chlorides of Na, K, and Ka, and that with rising volume rates, the milliequivalents of Na and K decrease, those of K remain constant. This suggests that while 9 can move independent of the shift of HK is secreted by all the cells. This result has led to the assumption that the parietal cells are the seat of the formation of the acid, that the non-parietal cells secrete Na and Ka, that both of them, parietals as well as non-parietals, equally eliminate K, and that the increase in parietal secretion is accompanied by a relatively small increase of non-parietal secretion. It will be seen that strong support to this assumption is available. The next question is as to how the amazingly great concentration of hydrochloric acid can be accomplished. This question can be reduced to the somewhat simpler problem of raising the hydrogen ion concentration to a level about 3 million times above the blood level. For it can be assumed that Cl, the concentration of which in the gastric secretion is about 1.6 times higher than that in the plasma, is passively dragged along into the parietal cells according to the principle of electroneutrality. Provided that the parietals are any impermeable, like the red blood corpuscles, definite proof of the parietal cells being involved in the production of HCl has been advanced by the discovery of Davenport that the parietal cells are the scat of large quantities of hydrascine discs of the blood-free mucosa are punched out of the stomach walls and cut into thin slices parallel to the surface. The enzyme concentration in these slices is determined by their catalytic activity and compared with the number of parietal cells contained in histological preparations of the corresponding slices. The result is a very high correlation between cell count and enzyme concentration. This concentration in cat cells being even 6 to 7 times greater than in the red corpuscles. The bearing of carbonic anhydrase upon the formation of HCl can be assumed to be this: CO2, which is continuously produced by the metabolic activity of the parietal cells, is rapidly hydrated to H2O3 at a rate corresponding to the high content of carbonic anhydrase, and HCO3, resulting from the immediate ionization, is exchanged with equivalent amounts of plasma Cl, while the ill moiety of the H2O3 molecule remains in the cells. This Kiel shift from the plasma into the cells would appear to be just opposite to what is happening with other tissue cells which are lacking in the enzyme. In these tissues, likewise, CO2 is produced but diffuses as such into the plasma and enters from there the red blood corpuscles. Due to the large content of enzyme in the erythrocytes, it is again catalyzed to H2O3, immediately ionized to HCO3 and H. And while H is fixed to the hemoglobin, HCO3 leaves the cells in exchange for entering. This is the normal, well-known shift of Cl from the plasma into the red cells. The reverse TL shift E Davenport from the red cells to the euasma in the blood passing through the gastric mucosa could be expected to take place, and has actually been found. Its occurrence is further indicated by the fact that blood passing through the secreting stomach becomes more alkaline due to the copious entrance of, whereas in passing other active tissues, muscle, it becomes more acid. Through these investigations, it became clear that the origin of the hydrogen ion moiety of the hydrochloric acid can be assumed to be to derive from the metabolism of the active parietal cells. The other moiety Cl being supplied from the blood plasma, but this remarkable advance in knowledge leaves unsolved the most essential problem, viz., the mechanism by which the accumulation of H to such tremendous height can be accomplished. The penetration of glands by acidic and basic organic substances. In view of the enormous accumulation of H in the gastric juice, there can be no doubt that this achievement is a matter of active transfer. There are many observations concerning organic substances, which, after intravenous injection, appear in the gastric juice in so much enhanced concentration as to be easily mistaken for real secretion, until a more careful analysis of the possible employca shows tluon to be only incidental effects of glandular activity. This refers particularly to dyestuffs, which, even with a relatively low accumulation ratio, give the illusion of a high concentration due to the intensity of their color. Observations concerning such false effects have been mentioned already in the introduction to this section, pages 525f. On the other hand, in the subsection concerning liver, such accumulation ratios for dyestuffs will be cited as to indicate by their large values, sometimes greater than 1, hero and unambiguous active transfer. Penetration of dyestuffs already several times has played a role in this section. Particularly kidney C experiments have provided evidence that, in general, the proximal tubules cannot be penetrated by acidic dyestuffs unless certain physicochemical properties based upon a non-polar-polar configuration of their molecules, probably leading to a characteristic interlinkage with the cell surface, are present. Moreover, as we will see later, the dyestuff transporting activity of the liver cells apparently is bound up with specific physicochemical properties of the dye different from those involved in the transfer by the kidney. On the other hand, from various more sporadic observations, one may conclude that in the intestinal mucosa, in the body surface of animals, and in the salivary glands, conditions favorable to dyestuff accumulation are absent. Somewhat different is the behavior of the gastric mucosa and of the pancreatic gland, the accumulating power of which appears to be connected with the acid and alkaline reactions of their secretion. Entrance of dyestuffs into the gastric and the pancreatic juice. DT-OGs were injected intravenously with numerous dyestuffs belonging to various chemical groups, and varying in diffusibility and lipoid solubility, in oxidizing and reducing power, and in adsorbability to proteins. The gastric juice was collected, and the appearance of dye studied. The result shows that the deciding factor is essentially the basic or the acidic character of the dyes. For instance, in the experiments of Ingram and Vischer, among 27 dyestuffs appearing in the gastric juice, all but one are basic, whereas acidic dyestuffs fail to appear, those dyes being called basic, the staining power of which is carried by addication, while in the acidic dyes the anion bears the color. The result is correlated to a corresponding series of observations concerning the pancreatic juice. Here, in the same type of experiment, mainly the acidic, anionic, but also the amphoteric dyestuffs under some conditions, were found to enter the juice. In general, the F effect is more evident with gastric than with pancreatic secretion, due to its more definite acid character compared to the weaker alkalinity of pancreatic juice. This is especially emphasized by the high accumulation ratio observed with basic dyes entering the stomach, the concentration of toluidine blue, for instance, compared to the blood being found to be 36.5. That of theanine 36.8, of neutral red 26.4. However, this result is by no means a sign of secretory activity. The dyestuffs are electrolytes, basic dyes being in general weaker than the acidic, and therefore, due to hydrolysis, present in the blood more or less as free bases. These free bases, as undissociated molecules, and especially as lipoid-soluble substances, can diffuse across the cells, Sec4. 13. And enter the gastric juice, where they are converted immediately into ions, which, in general, are unable to pass the living cell. Thus, the basic dyestuffs are captured in the stomach in amounts increasing with the acidity of the juice. Such simulated secretion is a common, widespread phenomenon. Sometimes its real nature is very obvious, sometimes veiled, especially when, as in the case of the entire organism, complications arise from the coincident intervention of several organs. One instructive example is seen in dealing with the excretion by the isolated frog kidney of the sulfonic acid dyestuff, arsenal red and the basic neutral red, if the kidney is perfused from the aorta with Kinger-containing NH4CC. The pH of the glomerular filtrate running along the tubules can be decreased from 7.4 to 6.3 due to membrane hydrolysis with uptake of the NH3 by the epithelia. If, under these conditions, neutral red is added to the Kinger solution, simultaneously entering the kidney from the portal vein, a very high concentration of neutral red is attained in the urine with an accumulation ratio greater than 40. When phenyl red instead of neutral red is applied, nothing happens, no concentration change occurs following the addition of NH4CC dye in case the kidney is perfused from the aorta with ringer made alkaline by the addition of nalcose. The course of the experiment is the reverse of the former, with the concentration of neutral red falling off, the accumulation ratios decreasing. After the supply of nalcose has been stopped, the ratios rise again. However, when phenol red is used, its concentration is very little affected by this change to an alkaline reaction. The reason is that phenol red is one of those dyestuffs which, because of their molecular structure, are actively transferred by the kidney rather independent of the existing pH, whereas neutral red passes by diffusion here as well as in the stomach. The difference is dramatically shown by the fact that even though the activity of the kidney tubules has been suppressed by cyanide or by urethane, the concentration ratio of neutral red, which is low during the aortic perfusion with nalcose, rises quickly after NH4CI has been substituted for the bicarbonate. The phenomenon of simulated secretion is not restricted to dyestuffs. It is met wherever weak electrolytes are distributed between fluids of different PLL. Therefore, it is of great importance in the field of drug distribution, especially as a main factor controlling the toxicity of alkaloids. For instance, it has been known for a long time that after intravenous injection, morphine hydrochloride is accumulated in large amounts in the stomach. The salts of other alkaloids, strychnine, cocaine, nicotine, directly introduced into the stomach of the cat or dog are not absorbed at strongly acid reaction, but are rapidly absorbed and therefore generally toxic after the gastric juice has been rendered alkaline. With regard to the various possible implications obscuring the pattern of distribution of weak organic bases in experiments on the total animal, 65. The following study with sulfonamides may have a special informatory value. Gastric juice under maximal histamine stimulation, P. 599 is collected from Heidenhain gastric pouches of dogs to which various sulfonilamide drugs are administered. Conjuring the concentration of these substances in the gastric juice and in the plasma, the concentration ratios are calculated. It appears from the following table that sulfanilamide and sulfapyridine are accumulated, the others not. Cone ratio juice PASMA, the following interpretation has been proposed. When sulfanilamide dissolved in plasma is allowed to pass across a membrane into an HEL solution with its concentration corresponding to that in the gastric juice, 0.1 fat has been reached. The concentration ratio cannot be expected to be 1 because sulfanilamide reacts with HEL from the saturation concentration in this HEL and in plasma 3,080 milligrams percent and 1540 milligrams percent respectively. The concentration ratio, solubility ratio, is calculated to be 2.6. This is the same value as found in the experiment. Consequently, 2.6 is not an accumulation ratio in the usual sense, indicating a glandular active transfer P525F, but it is like a distribution coefficient. The question arises why in the experiments with the other sulfonamide, identity of the two values is lacking, and the solubility ratio always is greater than the concentration ratio juice plasma. An explanation can be arrived at by following the changes of concentration of the sulfa drugs in the plasma. After known amounts have been injected intravenously, the drugs leave the plasma by diffusion. not only through the stomach wall into the gastric juice, but also into the entire body through a great variety of pathways, according to the rates of diffusion in water or in organic solvents and other factors. The rates of diffusion measured in this way are listed in the last column of the table. It appears that sulfanilamide is the fastest in passing across the tissues. Sulfapyridine is next, the others much slower. Thus, one understands that the distribution of sulfanilamide comes to an equilibrium, whereas the other drugs fall short of it to a greater or lesser degree. For the formation of pancreatic juice, disregarding again the enzymes, the chief function of the pancreatic gland is the elaboration of a secretion which is slightly alkaline, 708.5, owing to its content of L-carbonate ion, as the character of the gastric secretion is chiefly determined salivary gland by its high concentration of water, which makes this glandular product a strongly hypotonic fluid. The problems of these three performances seem to be related inasmuch as they bear upon the distribution of the most common components of the body fluid, 2,2-OC, and H-tofu. But the three glands have attacked their problems in utterly different ways. Concerning the pancreatic secretion, one main question is whether the bicarbonate ion is derived from the metabolism of the pancreas, as is the hydrogen ion in the gastric gland, or whether its origin is the blood serum. Mainly two kinds of experiments on dogs have provided evidence that the source is the serum. First, with highly active glands, the concentration ratio of total juice CO2 and plasma CO2 has been found to be 4 to 5. And after an intravenous injection of radioactive HC3, see radioactivity, is distributed between juice and serum in approximately the same ratio. Whereas in similar experiments regarding radioactive Na, the distribution ratio was second. If the metabolic KO2 were the source of the HTO3 in the juice, one would have expected an influence of sulfanilamide, which by poisoning the carbonic anhydrase present in the gland tissue would inhibit the transformation of KO2 oxidase. But even in the presence of exceedingly large amounts of sulfanilamide, the juice does not undergo an essential change in composition. The next question is how to account for the rise of the HCO3 concentration to a level 4 to 5 times higher than that of plasma. It has been found that the concentrations of Na and K are approximately the same in juice and in serum. This, together with the findings that an experimental change of concentration of these ions in the serum yields a corresponding change in the juice, allows the conclusion that the gland is freely pegmyl to Na and K. Further, with increasing rates, CDAI secretion, the alkalinity of the juice rises due to a greater concentration of L-FeCO, and simultaneously, the percentage of Cl decreases, the sum of IL-CoA and Han concentrations remaining nearly constant. The concentrations of ACO3 and Cl in the gland tissue correspondingly change in the opposite direction. These facts are interpreted as being due to a free permeability of the gland cells to NAK and HCO3, and to a poor permeability to TL. Therefore, has been elicited by secretin more HCO3 enters the juice and less Cl in such a way that electroneutrality is maintained. This may be explanatory of the concentration ratio of juice HCO3 to serum IOCO3 found during the increased glandular activity. Recapitulation of the above considerations shows that, according to present knowledge, the properties of these gland cells are rather UNDO. Disregarding the differences in rates of penetration, not only Cl being slower, but also K-engine phosphate, the active cells appear to be permeable to the main inorganic cations and anions. Correspondingly, serum and pancreatic juice are in osmotic balance, as are serum and gastric juice, P5 and 99. It will be particularly useful to extend the investigations to experiments with non-electrolytes. Studies concerning the permeability of the pancreas to dyestuffs have 5, the permeability of the liver, about 80 years ago, when only a small number of colored natural products were available for experiment concerning the pathways of dissolved substances through the living body. There was discovered the neat picture of the biocapillaries. which appears in the liver after an injection of indigo carmine, and which furnish striking evidence of a secretory activity of the hepatic epithelium. In more recent 243 H.F. Tucker and E.G. Ball, J. Bile. Shera, 139, 71, 1941. E.U. Still, A.L. Bennett, and V.B. Scott, AMJ Physio, 106, 509. 1933, have studied the gaseous metabolism of the gland by comparing the rate of blood flow and juice production and the O2 and CO2 content in the pancreatic artery and vein during rest and activity. The most interesting result is that at the onset of secretion produced by an intravenous injection of secretine, for several minutes the venous CO2 falls below the level of the arterial Q, whilst HCO3 appears in the juice. Studies of this phenomena was found that the bile capillaries likewise become visible as a dark blue network after the indigo carmine dissolved in ringer has been perfused from the abdominal vein through the isolated frog liver, and that, subsequently in a cannula inserted at the fundus of the gallbladder, a dark blue secretion can be collected, the color concentration of which surpasses by far that of the perfusing solution. Extension of these observations to other technical dyestuffs showed that many of them likewise can be accumulated in the secretion, but peculiarly enough, the visible network of the stained bile capillaries has so far never been obtained except with indigo carmine. According to Höber and Titiju, among the dyestuffs which appear in high concentration in the secretion are many well-diffusible acidic, sulfonic, and carboxylic compounds having very diverse chemical structures, partly lipoid-soluble, partly lipoid-insoluble. It appears that in its power to accumulate the dyestuffs, the liver differs very much from the kidney, which can differentiate between the non-polar-polar and the polar-polar sulfonic azodyestuffs, and can secrete sulfonic acid triphenylmethane dyes, which the kidney cannot. It has further been observed that basic dyestuffs likewise are secreted, but apparently to a lower degree, probably for the simple reason that, due to their basic character, these vital stains, CP pound 63, are retained and stored in the hepatic cells. Colloidal dyestuffs, significant for their low diffusion rates, either fail to reappear at all or pass only in small concentration. The great affinity of the liver to dyestuffs may be conceived of as being connected with their great adsorbability because of the prevalent nonpolar organophilic groups in their molecules. This would make it understandable that no accumulation occurs with dyestuffs, which show a predominance of hydrophilic sulfonate radicals. Another viewpoint is found in the correlation between the perfusing concentration and the accumulation ratio. Resembling the analogous correlation found in studies of dyestuff transfer, for example, indigo carmine was supplied in 0.05% solution, and the accumulation ratio was found not higher than pound to 6. However, when a 100 times smaller concentration is used in the experiments of Höber and Titiju, the ratio G-nan exceed 1, and with a dilution of 500 times, ratios of even several thousand can be reached. This group of observations can be interpreted as indicating adsorption, as will be discussed later with regard to the concept of carriers as transporting devices, CP 577, also P618. But in view of these amazingly high concentrations found in the liver secretion, the question must be raised whether water reabsorption in the liver tissues might not play an important role. This might be suggested with regard to the aglomerular kidney, P568, and still more to the surviving excised frog kidney, P567. As mentioned before, the frog kidney, which is perfused from either supply route by ringer containing a dyestuff adequate for SEN accretion by the proximal tubules, is indicated by a marked accumulation rate in perfusing fluid, for example, 30 times. Docs not show any coloration of the tubular lernina under the microscope because the lumina are too thin to allow a sufficient absorption of light by the enclosed dyestuff solution. The dyestuff concentration is the result of the glomerular influx, the secretory tubular delivery of dye, and the reabsorption of water by the tubules. With an excised kidney bathed in dyestuff ringer, The dye-stuff concentration resulting from the activity of the proximal tubules alone can be observed easily since the glomerular influx is absent, and absorption can proceed so that the color filling the lumina increases in distinctness until finally it reaches very high values, but that the secretory activity also plays an important role is shown by the fact that this behavior is displayed only in the J resonance of a secretable dye, and furthermore, that the picture does not appear if to the bathing dye-stuff ringer solution a narcotic or cyanide is added. The situation in the ringer perfused liver is decidedly different anatomically, since obviously there is no unit comparable to the glomerulus from which the secretion could be diluted, and physiologically, since inhibition of active reabsorption of water by a narcotic or by cyanide should be followed by an increased output of fluid in the gallbladder cannula, which does not occur. On the other hand, after inactivation of the liver by phenylaurethane, the formation of the blue network of the bile capillaries, otherwise following the injection of indigo carmine, fails to appear, Plattner, Locke, Sit, and also the entrance of the dye into the cannula is reversibly diminished or Stoji ped. The idea that adsorption might be involved in the accumulation of dyestuffs is further supported by the finding that with substances which, in general, are lacking in adsorbability, secretory concentration is not obtained when supplied to the liver at molarities similar to those effective with dyestuffs, thus xylose, glucose, lactose. Inulin, with molecular weights ranging from 150 to about 5,100, reappear in the secretion of the ringer, perfused frog liver without a significant change in concentration, as though having passed an inert filter, whereas dyestuffs, with a molecular weight of about 500, simultaneously perfused show a considerable accumulation ratio. Only the very large starch molecule fails to pass the liver. In view of these facts, the question arises whether the carbohydrate molecules smaller than starch pass the filter of the liver across the hepatic cells, or across the intercellular pathways. A similar alternative was encountered earlier concerning the transcellular or intercellular passage of salts across the epithelial layer of the intestine, CP 540. Not only from the viewpoint of general physiology, but also from that of liver function, it seems unlikely that the normal hepatic cells should allow the free penetration of the sugars. Although a similar conclusion could be suggested by another series of observations in order to investigate the chemical conditions of the secretory activity of the isolated liver, a great number of seemingly rather indifferent organic substances was tested for their influence upon the dyestuff secretion. After a deep coloration of the fluid in the gallbladder, cannula has been attained to of the perfusing dyestuff ringer solution is substituted effect that the dyestuff concentration in the secretion falls off rapidly, eventually to zero, and reappears after resumption of ringer perfusion. This inhibitory effect is produced by non-electrolytes and electrolytes. The main non-electrolytes are the disaccharides, hexoses, pentases, polyhydric alcohols, Ce-suelectric amino acids. Succinamide. The main electrolytes are the Ni salts of rather strong dibasic and monobasic aliphatic oxy and hydroxy acids, like gluconate, succinate, pyruvate, and others. All these substances are rich in polar groups, oil co, NHD2, and therefore more or less surface inactive, hydrophilic, and as such dehydrating or antidispersing. With respect to hydrophilic colloids, pages 296ff, 245f in general, the larger molecules are the more active ones. For instance, mannitol and succinate have a stronger dehydrating influence and a more vigorous liver effect than erythritol or glycolate. Organic substances have their physicochemical, as well as their physiological antagonists in substances which, on the one hand, are surface active, dispersing toward various hydrophilic colloids, starch, gelatin, lecithin, and on the other hand, are stimulants to the liver secretion, cholinergic action, unless they are administered in such high concentration or for such length of exposure time that the dispersing action causes disintegration, cytolysis, and irreversible loss of activity, CP340, Substances having such action are the sodium salts of the higher fatty acids, higher than Cs of a like acid, salicylic acid, bile acid, and others belonging to the groups of hydrotropic P. 336. Solubilizing nonpolar polar substances. The antagonistic properties of the two groups of compounds appear most strikingly in experiments in which the inhibitory effect upon the dyestuff secretion of the liver by a substance belonging to the first group is neutralized by the addition of a member of the second group, which restores the concentration of the dyestuff. Thus, the entrance and the accumulation ratio of the dye might be regarded as dependent upon a certain consistency of the superficial and of the internal structures of the hepatic cells. At the beginning of this section, active transfer by an organ, as contrasted with passive penetration, was defined as the performance of osmotic work, empowered by the expenditure of some fraction of the energy which is liberated in the metabolic reactions. One may supplement this definition by mentioning light as another source of energy. This is mainly significant in plant life, and its biological effects are ordinarily mediated through the products of p-hyatosynthesis, the metabolic reactions of which likewise provide the energy for active transfer. 1 ton. Energy supplied for active transfer. As yet, the information about the origin of energy utilized in active transfer is rather meager, but it may be reasonable to surmise that carbohydrates, as the most common source of energy, wherever physiological work has to be done, appear in this role also here, even though the substances transported uphill are virtually different in their chemical nature. Surveying at first all those substances which in the discussion of this section have been proved to be subject to active transfer, we encounter 1. Hexus is shifted by the proximal tubules of the kidney, P561, and by the intestinal mucosa, P5142, amino acids, presumably shifted at the same places, pages 550 and 562. 3. The metabolic end products, urea and uric acid, P572, migrating through the wall of the kidney tubules in the opposite direction from that of hexases and amino acids. 4. Organic sulfonic acids as dyestuff intermediates, and related compounds which are transported by the kidney, P563. The liver, P606, certain plant cells, P265. 5. Special inorganic ions, HEOs transported in the distal kidney tubules, H by the parietal cells of the gastric glands, pages 560 and 599. 6. Inorganic salts, or better, inorganic cations plus anions by the intestinal wall, P556. The gills of fish, P586. The frog skin, P589. The kidney, P559. And various plant cells, pages 590 of Firwa. Now, among these substances, several seem to be forced through the cellular machinery by means of energy derived from carbohydrate. This appears most conspicuously from microscopic evidence in certain experiments of Steward, which have been mentioned before P591. Normally, the cells of potatoes live in a state of low metabolic activity. They are dormant storage cells filled with starch. Placed in highly diluted salt solution and strongly aerated, they change into metabolically vigorous cells. Oxygen consumption and TO2 production rise, the starch granules disappear, and simultaneously osmotic work begins. Salts are transported and accumulated in the enlarging cell sap vacuoles. But the specifically physiological way of disposing of the potential energy of the carbohydrates is better revealed in the following manner. As is well known, carbohydrate is broken down stepwise in a sequence of intermediary reactions to the metabolic end product. These reactions are mediated by enzymes. The main steps are the formation of several phosphorylation products, of pyruvic and of lactic acid, and others ending with the final dehydrogenations and decarboxylations. This chain of reactions can be interrupted at various places by poisoning one or the other enzyme active in the concatenation of single reactions. The effect is a block in the normal carbohydrate breakdown, which must be followed by more or less impairment of the supply of energy indispensable in each kind of activity, which relies on the utilization of carbohydrate. But in case of such an event, the activity should be revived or restored by repairing the chain at the point of interruption. In other words, by providing the intermediary product, which, after addition of the poison, ceases to be produced. As a matter of fact, this conjecture has been verified most satisfactorily by employing, for instance, iodoacetate, which blocks the chain of reactions between the intermediates triosphoric acid and phosphoglyceric acid, and thus prevents the formation of pyruvics and lactic acids. Then the arrested transfer can be set going again by addition of pyruvic or lactic acid, just as the working activity of muscles and the irritability of nerves is reestablished by lactate after poisoning with iodoacetate dichichat. This has been proved in various experiments upon the kidney, the skin, and the liver. It has been mentioned earlier, P569, that the secretory action of the proximal tubule of the kidney can be easily studied on cystic explants of the mesonephros of the chick embryo, which, when immersed in a dilute solution of phenylarchial, takes up this acidic diastuph into the lumina and accumulates it there to a very high concentration. If enough iodoacetate is added to the dyestuff solution to paralyze the uptake of dye, this effect can be counteracted by addition of pyruvate, lactate, or succinate, the latter being one of the breakdown products appearing in the aerobic phase of the carbohydrate metabolism. But glycerophosphate has been found further. It has been mentioned before that the frog skin displays an active transfer in that it transports Cl ions against a concentration gradient. This is evidenced in previously described experiments in which the isolated skin is interposed between samples of the same frog isotonic ringer solution, P588.5. It has been known for a long time, Yachong, that the surviving skin is able to drive a current of water from its epithelial to its cirrhosis side. Now, the more recent detailed studies of Huff have revealed the facts that, first, during the shift of fluid, the Cl concentration diminishes on the epithelial and rises on the cirrhosis side, indicating that the transport of water has been accompanied by an additional transfer of Cl. And second, as evident from the table P589, the active transfer of Cl can be diminished by bromoacetate, which resembles and that the poisoning action of bromooctate can be counteracted by lactate. See experiment 5. Also, the dyestuff concentrating capacity of the isolated ringer perfused liver of the frog, P7, which is decreased by iodoacetate as well as by other poisons, seems to recover after additional provision of lactate. But finally, it might be objected that the toxic effect of iodo, or bromoacetate, and the subsequent detoxicating effect of pyruvate, or lactate, as stated in the aforementioned experiments, cannot be considered a safe proof that the various kinds of active transfer depend directly on energy supplied from carbohydrate breakdown. For iodoacetate is a rather dangerous substance, the administration of which is readily followed by a universal and irreversible impairment of cell vitality and hence of every kind of cell activity. Even though pyruvate and lactate have been found to restore the inhibited active transfer, this favorable influence might be only an indirect symptom of the revival of the cell energetics, which seem to depend quite generally upon carbohydrate metabolism, see also P614. Inhibition of active transfer can also be produced by chemical means, but without affording clear evidence that the chemicals disrupt the coupling between a specific metabolic process, like the carbohydrate breakdown, and the active action of the transporting mechanism. Such conditions are present during anoxia and during narcosis, anoxia, produced by cyanide, hydrogen sulfide, or an atmosphere of pure nitrogen, results in cessation of the normal activity, for instance, of the dyestuff secretion by the chick mesonephros, or of the uphill shift of Cl by the frog skin, or of the accumulation of sulfonic acid dyestuffs by special cells surrounding the huff. Pogier's arch, FDG. Ihysial, 236, 635, 1935, vascular bundles of plants. But this inactivation cannot be correlated with the influence upon a specific group of CTGGL constituents serving as the essential source of energy, but rather involves the entire metabolism narcosis, as known, for example, from experiments on the diastoph perfused isolated kidney or the liver, even more than anoxia, is a manifestation of the unspecific interruption of numerous cellular reactions, since the narcotics, because of their surface activity, are believed to attach, in general, to the cellular and mycelar structures, which are the site of the enzymes, and to interpose in this manner a barrier between the manifold enzymes and their no special source of energy of a chemical nature, has been suggested as yet as potentiating active transfer of urea and uric acid. In the kidney, another iris for studying the interrelation between the chemical turnover and tin, osmotic work of living cells is based upon measurements of the influence of temperature within a certain range of temperatures. One ordinarily observes an increase of work with an increase of T temperature. For instance, as shown in Table 50 itone, this can be interpreted as dependent upon a higher rate of diffusion of the substances entering the energy sweep-line system, or a greater reaction velocity in this system. In general, the temperature coefficients of diffusion referred to a temperature interval of 10 have been found to be 1.2 to 1.3. The temperature coefficients of chemical reactions 2 to 3, see Sec. 1. Chap 2, the Keough values calculated for the experiment in the table are about 2. That is, like in this regard, it is worth mentioning that the inhibition of phenol red transport by the tubular cells of the chick mesonephros, following poisoning with hydrogen sulfide, cannot be counteracted either by lactate or by pyruvate, LV Beck and R Chambers, J Cell, and Compi. The temperature coefficients of metabolic reactions, but often they have been found to be as high as 5. Below a certain TMJ aerature limit, the work performed by the system is practically 0. This is evidenced by the following experiments upon the mesonephros of the chick embryo, P569. The uptake and accumulation of phenol read by the explant can be inhibited completely for many hours by cold, 3 degrees to 6 degrees Celsius. After the culture has been returned to body temperature, appreciable amounts of dye appear within a few minutes in the lernina and are accumulated. However, when the culture is exposed to low temperature after the lernina have been filled with color, the dye requires hours to escape. Obviously, the uptake at high temperature is rapid, active, and unidirectional. The outgo slow and passive. This resembles the situation in the experiments of Collender and Holmstrom, p. 614, also 265, upon the influence of oxygen on the intake and outgen of diastuffs. Subsequent lack of oxygen, the captured dye escapes, although very slowly. Another example deals with the absorption of potassium halides by Nutella with light as the ultimate source of energy ET2 through the metabolic reactions of the products of photosynthesis, and lowering the temperature to 5 degrees causes. For example, Cl2 leak out of the healthy cells, but at room temperature the 2. The cellular devices for active transfer and their mechanism. When a chemical process has to be carried out in such a way that a greater or smaller portion of the energy liberated becomes available for work, for instances, for osmotic work, it requires some kind of machinery to bring about an orderly movement of the molecules taking part in the chemical process instead of an otherwise random movement. We are still very far from perceiving any such structural arrangement, even though, as long as cells, either living or fixed, have been investigated by the microscope, emphasis was laid upon the interpretation of their visible constituents in the light of a certain functional significance. More successful in this regard were the combined efforts of histologists and physiologists to design a mechanical model of a muscle on the basis, particularly of the newly attained knowledge about the physics of fibrillar structure, their physical chemistry, and the linkage of this with an orderly system of chemical C, further the observations of Osterhout concerning the uptake of K by Fa Oma, which ceases in the dark and evidently is kept going by the photosynthetic reactions A. G. Jacques and W. J. V. Osterhout. J. General Physiol, 1727, 1932. Physiol, 35, 967, 1933. Further, the experiments of I.E. Harris, J. Biles Shem, 141, 579, 1941, and W. Wilbrandt. Pfluger's Arch F. Diges Physiol, 243, 519, 1940. A ppn uptake of K against the concentration gradient by human erythrocytes and its linkage with glycolysis reaction. See Sec 7. In the field of the present chapter, the appearance and the variable location of vacuoles and granules and secretory cells at rest or in action at first suggested postulating forces pushing these bodies in one direction toward the lumen, where the enzymes or hormones or mucin or other substances produced within the cells are extruded. In addition, it seemed fruitful to consider whether the more simple substances, like single molecules, produced elsewhere and present in the cell surroundings could be taken up, then accumulated, transferred across the fly cell body, and thrown out in a similar way. However, this picture looks less promising, since there are observations on living cells according to which, during the shift of molecularly dispersed substances, or small molecular aggregates, transporting vehicles such as granules or vacuoles cannot be detected. Even with a high-power microscope, this was emphasized, for example, by, say, Chambers and Kempton in their observations upon the secretion of phenol red, the surrounding highly diluted solution. It appears to be dissolution in the visibly stained protoplasma and is expelled in a markedly inert taste concentration at the luminal side. Also, in conflict with assigning to the vacuoles an important role as carriers, is the observation that, in the excretion of dyestuffs by the kidney of mouse or frog, the high tide of elimination is already past. Before dyestuff vacuoles appear under the microscope, however, there are reasons to believe that the vacuoles containing the dyestuffs are formed at first in submicroscopic sized liae, and intraplasmatic secretion, thus producing the appearance of a monophasic homogeneous distribution inside the cell, and that they grow only after a time to visible bodies. Hence, this discussion as to the role of vacuoles as essential parts of a transporting machinery remains inconclusive. Another source of information has been furnished by following the absorption of hexaces in the intestine and their reabsorption in the kidney, pages 544, 561. As was mentioned earlier, Wilbrandt and Last, on the basis of their finding that the differential absorption of the hexaces in the intestine disappears after poisoning with iodoacetate, suggested that the preferential absorption of glucose is tied up with reaction between the sugar and inorganic phosphate. Lunsgaard and Wertheimer showed that the preferential absorption of glucose by the intestine also is abolished by florazin, as has long been known for the reabsorption in the kidney. And Lunsgaard further found that in tissue extracts, florazin can be a strong in Kritzler, and Gutrunet succeeded in demonstrating in microscopic slices that the proximal tubules of the kidney, in which the sugar reabsorption takes place, are extremely rich in phosphatase, which is largely concentrated at or near the luminal border of the epithelium, and which at alkaline reaction can liberate from glycerophosphate. Inorganic phosphate, which becomes visible by precipitation in the tissue slices. They further showed that this dephosphorylation fails to be inhibited by florazin. These various separate observations can be linked together tentatively into a fairly reasonable picture of sugar absorption when the influence of is taken into consideration. First, dephosphorylation of glycerophosphate and hexosdiphosphate, which was mentioned as going on at alkaline reaction, DL7 to 9.2, and also in the presence of florazin, is inhibited at PTW6 to 7 by florazin. Second, the inverse process, the phosphorylation of glucose likewise, is inhibited by florazin at PLL5 to 7.4. Finally, taking into account the earlier mentioned fact, P. 550, that phosphorylation of glucose is an atopic reaction, viz., that it is a mechanism by which, in the presence of the adenylate acid system, oxidative energy can be utilized in the cell. The absorption can be described as an ACTA transfer as follows. At the luminal border of the epithelium, the PVL conditions in the phage-horalizing system of enzymes may be such as to handle the aerobic formation of hexocephosphate, as well as its entrance into the cells. There, at the protoplasmic reaction of about 7, dephosphorylation takes place, even in the presence of fluorescein and the freed sugar, which was shifted as hexocephosphate across the boundary separating the exterior and the interior of the body reappears. This hypothetical description of intestinal and renal transfer of glucose certainly is not more than a fragmentary picture, taking into account the following formerly mentioned findings. First, the secretion of diodrast reason to assume that diodrast is phosphorylized. Third, the localization of phosphatase at the brush border of the renal epithelia, which is suggestive of being instrumental in absorption, loses its meaning in secretion that starts on the other side of the cells. Therefore, the conclusion has been drawn that the enzymatic phosphorylation of sugar serves not only as a specific share in the mechanism of the transfer of sugar, but also as a more general source of oxidative energy furnished by the previously P550. This is somewhat similar to the reappearance by resynthesis of fat in the intestinal epithelia near their striated borders after it has been subjected to a transitory saponification, which provides the conditions for passage of the short distance between the lumen and the cell body, mentioned intracellular linkage between the carbohydrate and the adenylic acid systems, and as such cooperative in the shift of various substances. This energy output also may be thought of as being increased by thyroxine, the localization of enzymes, as it might set TV for the transport of substances across the cells. Constitutes a chemical organization comparable to the morphological organization, which was discussed before with respect to the cell vacuole, yes, and may be effective teosoulier as well. Thus, carbonic anhydrase has been mentioned as engaged in the accumulation of HCl in the gastric juice, P601, and in the reabsorptive shift of Icos in the distal tubules of the kidney, P560. However, so far, nothing of importance is known either concerning the structural organization of the parietal cells of the gastric glands as the site of the enzyme, or concerning the spatial relations between the specifically active tubular segment and the place where the enzyme acts. Other elementary components of a cellular transferring machinery, which have been mentioned previously, are the so-called carriers, hypothetical structures inside the cells, which have been suggested chiefly by the observations of MacLeod and McGeege, P546, Vuzar, P547, and Donhofer, P547, regarding intestinal absorption, and by those of 11, Smith and Shannon, P576, regarding kidney function. These and other observations have revealed the fact that the total amount of the substance migrating across the cells is composed of two fractions. One is the result of diffusion, and so increases with rising concentration of the substance. The other increases only in a certain range of low concentrations extending from 0 to a limiting value, beyond which it becomes constant, Sec figure 62, P577. This has been reasonably conceived of as indicating a system of intracellular carriers or vehicles, which, in the presence of a certain concentration of the available substance, are filled to capacity, and which subsequently discharge their entire burden to Gellner with the fraction pass things, mainly upon a special affinity between the substance in question and the carrier system, with the result that the total concentration in the output is raised in comparison with that in the entering fluid. Many questions are suggested by this concept, first regarding the nature of the affinity between the carrier and the load. The carrier may be looked upon as an adsorbent or as a solvent, or as a chemical substrate undergoing a stoichiometrical reaction with the transported substance. There are several properties of the transferring systems particularly favorable to the idea of adsorption. In the case of the kidney, among a great number of lipoid and soluble organic electrolytes. So far, mainly those have been found to be picked up and to be accumulational, which have a non-polar-polar molecular configuration, P566. This has suggested the interpretation that the nonpolar part of the molecule, due to some residual valence forces, attaches to the organophilic elements in the cell body, leaving the polar part oriented toward the adjacent aqueous phase. A symmetrical hydrophilic structure, or a predominant influence of the hydrophilic character, is detrimental to the active transfer, and presumably to adsorption. The behavior of the liver presents a somewhat different aspect. P. 607. The marked influence of the location of nonpolar and polar groups in organic electrolytes found in the kidney studies, for instance, with lipoid-insoluble diastuffs, fails to be manifested by the liver. Rather, all kinds of diastuffs are secreted except those which are highly colloidal, and those which contain in their molecules a great number of the strongly hydrophilic sulfonate groups. On the other hand, lipoid-insoluble organic substances like xylose, glucose, and lactose pass the surviving liver like a dead filter, without any change of concentration, P608, and inulin MW equals 5,100. Even under conditions as nearly normal as possible, that is, after intramuscular injection to the aglomerular toadfish, P568, reappears in the secretion of the liver at an average concentration of 0.57. Compared to a serum concentration of 1 nm, it is tempting to believe that this different behavior of the two groups of substances toward the secreting system of the liver is accounted for by the great adsorbability of the dyestuffs. In contrast to the various carbohydrates, which are unsuitable for adsorption because of their highly hydrophilic character, adsorption likewise might be revealed as an essential factor of the active transfer by quantitative studios, such as, for instance, that graphically reproduced in Figure 6J. 577. Reg resenting the excretion of phenyl red by the frog kidney. In this figure, the tubular action, as related to the concentration of diastuffs in the perfusing fluid, appears as a curve, the slope of which is initially steep and gradually swings to a plateau, interpreted as representing the maximum load of the carrier. The curve resembles an adsorption isotherm, with its increments decreasing with increasing concentration until an adsorption maximum has been attained, possibly indicating a saturation of the surface by the adsorbent. The figure can also be explained by thinking of the carrier system as a second solvent dispersed in the aqueous phase of the cell, the substance to be carried being distributed between the two phases, with a distribution factor decreasing with increasing concentration. Such a situation obtains when the substance is present in the two solvents in different molecular states, for instance, single molecules in one solvent bimolecular, in the other, see Chap 5, Subchap 9, and Chap 23, Subchap. In supplement to the discussion of the hypothetical carrier system, it should be said that the energy required by the system would be utilized for various purposes: for the loading process, for shifting the carrier across the cell, for setting free the substance from its attachment, and for its elimination from the cell. Further, it may be added that the unidirectional shift seems to be predetermined, in some cases, by structural components of the protoplasm, like the filamentous mitochondria providing, through their arrangement, intracellular channels parallel to the long axis of the cell. But, in addition, there is an intracellular polarity demonstrable in the kidney epithelia by a characteristic arrangement of vitally stained granules, which is destroyed when the cells are isolated, and when simultaneously they lose their ability to transport and to accumulate the phenyl red. 3. Mechanism of the simultaneous active transfer of cations and anions. Purn. This extremely fragmentary description of some separate elementary constituents of the transferring machinery, we finally turn to the most difficult problem, namely, to explain the fact that, by means of an appropriate setup of these elementary units, the energy liberated by metabolic reactions may ultimately serve to bring about the mechanical effects of active transfer, accumulation, and forwarding of the substances in question to the terminus of the process. In discussing this fundamental problem, first, some simple model experiment will be recalled, which could possibly shed some light upon the transport of the most common components of living organisms, inorganic ions, and water. In the introduction to this section, P5 or I6, studies were mentioned concerning the spontaneous movement of ions against the concentration gradient by employing a selectively ion-permeable membrane, for instance. In the following way, a cation-permeable membrane separates two salt solutions, each of them containing two K ions in concentrations such that HO and K equals CO, the kind of anions being irrelevant. Then H will tend to move from inside to outside, but for the RTASIN of electroneutrality, it cannot do so, unless for each H leaving, 1K enters against an ever-increasing concentration gradient. Equilibrium is reached. Eventu ali after a long time, when HO equals Kiko, for example. In an experiment of Netter employing a dried collodion membrane, P520 after 14 days, that is, before the final state was reached, XO equals 10. In a Corre sponding experiment with a selective anion-permeable membrane and the anions HEO3 and GL, the equilibrium would be readied if Eche Casticoso equals KOTTT. It was mentioned previously that the establishment of similar membrane equilibria has been observed under physiological circumstances, for instance, with muscle fibers, where, disregarding several complicating factors, Chappie. 17. The high percentage of K, compared to the much lower percentage in the serum, can be referred to their specific permeability to cations. However, the quotation of these observations seems to be without informatory value for the present question, since in the transport of ions others, we are dealing first with the simultaneous or approximately simultaneous accumulation of cations plus 8, and second, with processes demanding the liberation of energy, whereas in the model experiments, the concentration gradients arise spontaneously, and either for anions or for cations, due to the permeability of the membrane, which is either anion or cation permeable. At first sight, then, it may appear promising to adjust the model experiment to the exigencies of the physiological accumulation of both cations and anions by simply producing artificial membranes, which have a mosaic structure, that is, which are composed partly of each impermeable and partly of impermeable areas, each of them independently accumulating cations or anions respectively. For instance, with H and ICO 3 on one side, K on the other side of the mosaic membrane, corresponding to the aforementioned experiments, K and GL, would be expected to be shifted across the membrane against their gradient. A mosaic membrane can be prepared aging on a mercury surface by placing close to each other droplets of alcohol ether solutions of collodion and of rhodomin collodion, P317, which, flowing taut thunder and drying, form membranes with the suitable J properties by measuring the potentials across such a membrane interposed between two different salt solutions. The membrane can easily be shown to exhibit the expected behavior, as is evident from the following table. The figure pictures on the left side one Eschen permeable area, the substance of the membrane being electronegative, on the right side. One Aeon permeable area, formed by an electropositive substance, the two areas electrically influencing each other by way of the two U pieces, just as adjacent areas of a mosaic membrane must. When the entire membrane is in contact with the 0.1 mole KCl solution on one side, with the 0.01 mole KL solution on the other side. The dotted arrows in the figure indicate the direction of the transport of positive K and of negative Gl ions, respectively. The solid arrows the direction of an electric circuit, current with the membrane potential on the left side, U noting the cations on the right side, the anions in equivalent amounts from the more concentrated to the less concentrated solution, so that cations and anions neutralize each other. It is obvious that the mosaic membrane, at variance to the first assumption, must allow or even accelerates the leveling out of the concentration gradient of both cations and anions, rather than precludes an accumulation of salt. But nevertheless, it does not seem quite futile to utilize the idea of a mosaic membrane in an effort to adapt the model better to the physiological circumstances. First, Allowance must be made for energy derived in the case of physiological material from metabolism. In addition, it should be considered that natural plasma membranes, instead of being built up by such inflexible material as collagen, actually are composed of many substances of great diversity of physical and chemical nature, and so are not unlikely to possess mosaic properties. These points have been particularly, as to the first point, Brooks has proposed the following way to introduce the factor of energy supply into the exploration of ion accumulation in model experiments. The inside of the mosaic membrane is placed in contact with two NH-CoH3 ions, the most common products of respiratory metabolism continually arising inside the cell in excess, and therefore tending to pass the membrane. The ions across the cation permeable areas of the membrane, HEO3, correspondingly across the anion permeable. In addition, K and ions are distributed from the beginning of the experiment inside and outside in approximately equal concentrations, analogous to the formerly PT 120 described experiment of Netter. Then, if single selectively anion and single selectively cation permeable areas were electrically segregated from each other, simultaneously with the escape of H and HCO3, K and KL would be accumulated inside. But under ordinary conditions, that is, without such insulation of the cation and anion permeable areas, such an accumulation of K plus TL could not persist because, according to Figure 64, it would be annihilated by the electric currents. However, the question can be raised whether a continuous and abundant supply of metabolic H and ICO3 would not overcompensate the return of K, and thus produce a steady state of accumulation. As to the second point, the complexity and the flexibility of the plasma membrane, the evidence presented by Stewart and Hoagland should be reconsidered, page 592 theft. They found, especially with reactivated potato discs and with growing barley roots, that with rise and fall O2 consumption and CO2 production. In other words, with corresponding changes of H and HEO3 concentrations. The accumulation ratio for salts increases and decreases eventually to zero, that the accumulation of Cl, evoked at first in Nutella by illumination, vanishes again in the dark, which renders O2 unavailable, Sean 21. Further, the rate of accumulation is high with Cl Br Nas, low with E is 4, and high with K, lower with an A, and still lower with Tn. This would be in agreement with the numerous observations on artificial and natural membranes, showing that the penetrating power of Cl surpasses that of SO4, and that of K is greater than that of K hot. In addition, it has been observed that the rate of metabolism also depends upon the ions present, the metabolism being stimulated by K, depressed by C8, and also variously modified by anions such as WG or Clee's findings, may be accounted for as owing to the structural flexibility of Tli natural plasma membrane, mediated by its colloidal components, and at variance with the behavior of the inflexible collagen membrane in the model experiment. Further, such a flexibility would possibly be signified by a deformation of the pore diameters in a sieve structure, also to a change in the thickness of the membrane, both of which would produce an increase or a decrease in the potential drop across the membrane. 4. Inadel experience concerning the active transfer of water. The irreportant problem of active X transfer of water against its concentration gradient weakly has been touched upon on many occasions in the previous discussion, especially in connection with the transport of solution by the intestinal wall, by the kidney tubules, and by the various membranes of the body surface, may seem to have been unduly neglected so far. And also, here the discussion will be confined to the description of model experiments dealing with active transfer of water through electrical forces developed in artificial membranes and manifested as electroosmosis. Physiological transport of water in general is a very complex phenomenon, the thorough analysis of which leads to the distinction and evaluation of a series of factors. In part conceivable as a passive penetration along gradients of hydrostatic or osmotic or colloidosmotic pressure, also controlled by the diffusion rates of the solute. In part controlled in a somewhat undefined manner by the action of endocrines, especially the pituitary gland and the adrenal gland, but in part also definitely a clear-cut active transfer by glands, where the secretion pressure has been found to exceed the arterial pressure. This became especially obvious in studies upon the Eigelmüller kidney, P569, further P570, where the ureteral pressure seems to be 4 to 5 times higher than the pressure in the kidney capillaries. Possibly the physicochemical conditions for such an accomplishment are comparable, to some extent, to those established in models arranged to illustrate anomalous osmosis. During the last 3 decades, it has become increasingly obvious that the phenomenon of anomalous osmosis, discovered about 100 years ago, forces acting in normal osmosis. This could be anticipated already from numerous observations by Thomas Graham and others that anomalous osmosis in general is bound up with the presence of electrolyte solution. In anomalous osmosis, the fluid movement across the pores of a membrane separating an electrolyte solution and water, or 2 electrolyte solutions of different concentrations, is either unexpectedly great or small with respect to the osmotic strength of the solutions, or is Van versed in direction resulting in negative osmosis. More specifically, it was found that with solutions of equivalent concentrations, the rate as well as the direction of osmosis differed from one electrolyte to another. According to the investigations of Girard, Bartel, Loeb, Freundlich, Solners, and others, 2 factors are of outstanding importance. First, the liquid junction potential, E potential, between the 2 solutions which are in contact with each other across the pores of the membrane, and second, the electrokinetic potential, F potential, between the capillary walls of the membrane and the movable solution filling the pores. The height and the direction of the E potential vary according to the electrolyte concentration and the nature of the electrolyte, particularly its transfer numbers U and V in free solution, to the sign of the charge of the pore walls and the charge density, and to the size of the pores. These factors, in their combination, allow the ions to penetrate freely the membrane, or restrict their movement toward a selective cation or anion penetration, P317. The F potential depends upon the electric charge due to the nature of the membrane material and upon the relative adsorbability of the cations and anions of the electrolyte present. The cations being more adsorlaxal by a membrane carrying a negative charge, and the anions by one carrying a positive, and polyvalent ions having a stronger influence on the IE potential than monovalent ones, CP303. The result is the formation of a Helmholtz double layer, whereby the charges of the one layer are seated in the surface of the mobile fluid vein, so that due to the E potential across the membrane, and according to its direction, an attraction or a repulsion is exhibited toward the fluid vein. This, however, in itself is not enough to bring about an electroosmotic liquid movement. Electroosmosis requires the continuous flow of an electric current that means the existence of a closed circuit. Therefore, if electroosmosis occurs, a closed circuit must exist. If all pores would be electrically identical, then obviously no current could flow. However, if different pores have different electric characteristics, for example, due to their different sizes, then they yield different potentials. These potentials, according to Michaelis, may vary from the free diffusion potential to the membrane potential found with selectively anion or cation all-permeable mem act upon each other, and small electric arc currents must circulate through neighbor. In pores, a higher electronegative force of plus or minus one pore enforcing an electroosmotic fluid shift in the other. And in a direction, dictab the CT, the situation is typictai in Titani schemes. I positive in neolois, osino da cis. Figure 5, conferring the theory of positive neon electrical double layer, whose charges are partially fixed by electrostatic forces, partially movable. The potential across this pore is positive and high due to the mobility of H ions plus power. The lower pore permits penetration of anions as well as cations. The potential has the same sign as in the case of the upper pore plus 6, but is lower. That is, it approaches that of free diffusion. Therefore, since plus E is greater than plus E, a driving force exists which compels the liquid vein with its electropositive surface to move in the direction of the positive current dotted arrow, and thus increases the conventional osmotic flow associated with the concentration difference. This is NO-Fi as positive anomalous osmosis. Figures 66 and 67 picture eases of negative osmosis. According to some further comments are required regarding details, which have been or will be found to be important for further experimental progress in this field. First, anomalous osmosis is associated with the presence of appropriate electrolyte concentrations, as appears clearly from the well-known figures of Loeb, illustrating his experiments, double-endothelial collagen membranes. This effect, A proximately accipial to EXF, can approach zero, as E with low concentrations, F with high approximate zero. In many experiments, concentrations between M16 and M108 have been found to give gratifying results. Beyond that range, only normal osmosis occurred. Second, in securing clear results very much depends upon suitable membranes. In the case of collodion membranes, enormous differences in electrical properties can be produced by activation through certain chemical manipulations. Furthermore, the rigid collodion membranes can be better adapted to physiological purposes by impregnating or coating with collodiaphilites, gelatin, albumin, hemoglobin. In this way, they become sensitized to the influence of ions, particularly of ions which, like H, or like certain polyvalent anions, are products of metabolism. Thus, the F potentials can be changed, and indirectly the E potentials, the latter also eventually being decreased or annihilated at the isoelectric point of the collodiaphilites. For instance, the protamine samine with an isoelectric point of PGL12 to 12,4 is useful for preparing very active electropositive collodion membranes, which maintain a definite positive charge at acid reaction. And even at the alkaline reaction, pH 8, colloid amphilites, due to their property of shrinking and swelling, may also change the pore diameters according to the ions present, and thus modify the anomalous osmosis. Third, of greatest significance for the physiological problem in question, that is, the transport of water, considered as a positive or a negative ashechu osmosis, is the time factor. It has been noted by Loeb and others that, with a series of different electrolytes, the optimum time for observing anomalous osmosis with ordinary collodion membranes, such as he made use of, is about 20 minutes after the beginning of the experiment. Later, the phenomenon becomes less and less conspicuous due to diffusion of the electrolyte across the membrane. In addition, the klectroosmotic IC current, if it is directed along the diffusion gradient, contributes to equalization of the concentrations on either side of the membrane, since a shift of ions is necessarily associated with TLNG, electroosmotic shift of water, as in the previously discussed case, P622. There arises again the crucial question of the accumulation of ions, that is, whether such model experiments are of any value, as long as it has not been satisfactorily determined how or whether the electroosmotic shift of water can be made continuous by a metabolic energy supply. This problem will be discussed after reference to one of the few quantitative experiments which have silicate were formed in parchment test tubes, annual. Use osmosis. They were filled with 0.01 ml liquid and fixed in a beaker containing 0.1 m liquid. The electrical charge of the membrane substance is negative. The width of the pores in membrane T is large compared to those of membrane incipient. Then the behavior corresponds to the scheme in figure 66. The I potential of I is 16 MV of 2, near to 0. Therefore, a positive electric current circulates from inside 2 by way of the agar bridge to inside I, and back outside from I to 11 solid air. Further, potential I being greater than 2I in electroosmotic current, a positively charged fluid is forced through 2 from outside to inside, indicating by a rise of the meniscus in capillary 11 a negative osmosis dotted air. The rise in the experiment was not more than 4 millimeters, due to several unfavorable, though well-controlled, conditions, chiefly to that instead of one membrane, containing closely spaced pores of different widths and thus influencing each other over a very small distance. two membranes had to be employed, one providing the electromotive force, which drove the fluid by electroosmosis through the other distant one. But the position of the meniscus was found to be constant for more than 1 hour, and when it finally fell, it quickly regained its level after the beaker and the tubes had been refilled. That is, after restitution of the concentration gradient, which otherwise vanished quickly at the wide membrane hyphen. That the maintenance of the position of the meniscus is due to the circulation of the electroosmotic current was proved by the fact that the meniscus fell immediately if the current was interrupted by closing the agar bridge at its left end. Returning now to physiological studies, it may be recalled that 70 years ago, Engelmann had suggested that fluid transfer in absorbing and in secreting organs is of electroosmotic origin. Weaver, about 20 years later, this hypothesis was rejected by Reed Assis on the basis of his own well-known experiments upon the shift of water across diaphragms of surviving frog skin and mammalian intestine P552. His main objection was that purely secreting membranes often exhibit electric currents in the same direction as the absorbing membrane. But in the meantime, much progress has been made, particularly concerning the electrophysiology of the excised frog skin, which has rendered promising an effort to correlate the electrical and the mechanical properties, water transport, of this material, which, by its stability, provides better conditions for such a task than any other material. The resting potential of excised frog skin, mounted between two samples of Ringer solution and provided with oxygen, can be maintained fairly constant over several hours, the inside being electropositive, the outside negative. Hypoxia or anoxia decreases the potential, often reversibly. The same effect is brought about indirectly by adequate doses of KCN, Na2S. Or therefore, the normal frog skin potential can be believed to be the result of the respiratory metabolism of the living organs. This conception has been substantiated by investigations of the influence of bromoacetate, lactate, and cyanide, with bromoacetate resembling iodoacetate. The normal carbohydrate breakdown is interrupted in such a way that the production of lactate stops. This does not affect the potential so long as the supply of lactate in the skin remains. However, when it is exhausted by tissue oxidation, the potential falls off but can be restored of the presence of lactate, O2, and bromoacetate. It may be concluded that oxidation of sugar is somehow connected with the electrical activity. Regarding the need of energy supply for keeping up the potential, it should be mentioned that various narcotics known as depressants of metabolism have been found to decrease reversibly the skin potential even to zero, but not only the potential of the skin is dependent upon oxidation of sugar, but also as followed from the corresponding effects of the aforementioned chemicals. Its mechanical activity in pushing water from outside toward the body fluid, even against a hydrostatic pressure of 2 to 4 centimeters. She treats simultaneously with the water. A shift of Cl occurs, which, as noted in pp. 509, is influenced by the same compounds as the shift of water. Now, in order to analyze this machinery, its components must first be located in the various strata of the skin. In general, it is believed that the layer of surface epithelium is the active part. In particular, the electrical activity has been assigned to this region. According to Amberson and co-workers, the frog skin potential is the sum of 3 potentials. One located in the outside layer is the skin potential proper. It disappears when the skin is killed by placing it for a short time in an acid solution, about PVL4. The other 2 are observed with the dead skin when it is LACD in 2 salt solutions of different concentrations. One of these is a simple diffusion potential. The other is like the potential of a protein membrane, which above or below its isoelectric point, about P ratio of 4.5, is more permeable to cations or to anions, P319. Other reasons for assuming that the layer of the living epithelium is the active part are based upon the oft-noted observation that in order to AFCI the potential with ions or narcotics, smaller doses or a shorter exposure are required to bring about the change from the outer surface than from the inner surface. Thus, in summary, it appears first from the physiological experiments that a correlation exists between the transport of water, the electrical activity, and the metabolism. Second, from the physicochemical studies that a model membrane of a certain porous structure, bathed in two electrolyte solution, has the power to produce electromotive forces, which can drive an electroosmotic current across the membrane as long as the two electrolyte solutions do not reach diffusion equilibrium. As a matter of fact, the last described model experiment, P. 627, has shown that the electroosmotic transport can be maintained if care is taken to re-establish the decreasing osmotic imbalance. Therefore, the essential physiological problem is how, in the surviving skin, the persistency of the osmotic imbalance is guaranteed. A preliminary solution may be sought in the following, which is similar to a previously proposed answer, P. 622. The skin epithelia are the site of oxidation of sugar, and therefore the site of a permanent production of H and ions. A concentration gradient of these ions directed from the interior of the cell to the exterior can be visualized as permanent if removal of the ions is ensured by the tissue fluid in the deeper strata of the skin. The epithelial layer, then, may be pictured, for example, plasma membranes of the epithelia, the narrower pore to the utericigular cementing substance, the right side of the scheme representing the deep or subepithelial layers of the skin. The left side, the metabolizing stratum of the epithelial bodies. Then, the higher potential plus GJ due to WN1 would indicate the positive potential ordinarily let off from the inner surface of the skin, which causes the negative osmosis to go from the outer surface of the skin toward the inner surface. Of course, this is not more than a rather arbitrary and fragmentary approach to an understanding, but perhaps comparable to the more specified approach of correlating chemical and mechanical work in muscle, which has been mentioned before. 5. Supplementary remarks concerning the action of the choroidal plexuses and the ciliary body. Although a great amount of work has been done in order to clarify the forces involved in the production of the cerebrospinal fluid and the aqueous humor, no final standpoint has been attained. However, it appears highly probable that secretory action is responsible for the formation of these products by the choroidal plexuses in the brain and the ciliary body in the eye. This assumption is supported of the blood fluid. Compared to its ultrafiltrates and to the fluids in question, the gradients of capillary colloidosmotic and Donan pressures, the penetration of these organs by solutes, particularly by diastuffs, the histology of their surface epithelial cells, and the changes in their vacuoles and mitochondria, which follow the injection of P-arosympathetic drugs, the respiratory metabolism, and the effect of metabolic poisons, to the small size and irregular form and the complicated vascularization of these organs, and to their extreme sensitivity to insults, especially in the case of the eye. In the experiments, which will chiefly be referred to here as a supplement to our other discussions about active transfer, a simplification of the experimental conditions has been attempted by studying the freshly excised organs. The yashirari body was removed from the enucleated eye of rabbits, guinea pigs, and cats, the choroidal plexuses from the brain of pigs. Both prepa rations were laid flat, a glass plate, and moistened with ringer, and the distribution mainly of basic or acidic dyestuffs was studied under the microscope. Their solutions were provided to the stroma directly or indirectly by a preceding intravascular injection, and to the epithelium by placing the edge of a piece of tissue in such a way on the glass that the epithelial cells could be brought in immediate contact with the dye solution. With both preparations, the authors came to the conclusion that the basic dyestuffs move in the surviving organs from the stroma, across the basal membrane into the epithelial cells, the acidic dyestuffs in the opposite direction. Or in other words, the colored cations pass in one, the colored anions in the other direction. But it must be said that the short description, omitting details, leaves some doubt as to an unquestionable demonstration of this shift. The satisfactory condition of the material seems open to question in view of the low viability of mammalian secretory epithelium. Moreover, it seems hardly possible not to encounter difficulties in the interpretation, arising from the strikingly different staining power of L-acid and acidic dyestuffs in general, which are the result of the greater adsorption of basic dyes to proteins, and the lipoid solubility of the basic contrasted with the minor importance of this property in acidic dyes. In addition, the individual dyestuffs of each group differ with regard to the more diffuse or more differential distribution inside the cells, and they differ with regard to their capacity to permeate the living cells. If the interpretation of the microscopic localization of the dyes as indicating an opposite movement of cations and anions be adopted, it appears a reasonable conclusion of the authors to postulate an electric force not only to distribute the dyestuff ion, but also to establish an electroosmotic stream of fluid into the ventricles of the brain and into the anterior hemisphere of the eye bulb, respectively. In searching for the source of energy, the respiratory metabolism of the choroidal plexus and that of the ciliary body were studied by estimating the free energy of oxidation, or, more specifically, by determining the oxidation reduction potential with a series of redox indicator dyes, which were introduced into the stroma and the epithelium of the tissues, and the bleaching of which was observed under the microscope. Under aerobic conditions, the redox potential in the epithelium of the ciliary body, for instance, was found to be plus 0.1 volt, in the stroma 0.1 volt, referred to at pH 7. Nitrogen, the potential difference between epithelium and stroma disappeared, for instance. The potential of both epithelium and stroma decreased to 0.29 volt each, as indicated, for example, by the beginning reduction and decoloration of injected safranin. But after, several years ago, on the basis of a correlation between the biopotentials of plant tissues and their respiration, E.J. Lungead had come to the conclusion that the currents drawn from the surface of plant roots are due to oxidation reduction potentials. Such interpretation often has been rejected for the reason that an oxidation reduction process is accomplished by a transfer of electrons, and electrons happen to be conducted only along a metallic pathway, but not along a non-metallic pathway, like a physiological membrane, which, like water, can be a carrier only of ions, see Sec 6, Chap 26. However, it also has been questioned whether this objection is valid for all distances, since obviously it is not true within molecular distances, and for this reason, more recent studies regarding the conductance of artificial precipitation membranes, which so frequently have served as models for many kinds of natural membranes, may demand special attention. In 1867, Becquerel discovered the following phenomenon. A test tube, which has received very fine cracks, is filled with a solution of Q2, N3, and immersed in a solution of Na2S. After some time at the inner surface of the tube, there appear precipitations of metallic copper, and at the outside the liquid becomes dark yellow from the formation of polysulfides. In other words, at the inside reduction, at the outside oxidation takes place. According to Freundlich, Ostwald has interpreted the phenomenon in the following way. The solutions are separated by a film of QS, which is formed in the cracks, and which is impermeable to and SS. At the outer surface, S is oxidized, 2S equals S plus the liberated electrons, E pass the film and bring about the reduction. Electron neutrality is guaranteed by the film being permeable to no and not. So far, this hypothesis may be compared to the ordinary conception of the effect of an oxidation reduction system, which provides an electric current of ions. When two solutions of different oxidation reduction potentials are separated by a diaphragm, a membrane of a noble metal, PTO, and by leading off in the usual way with saturated KTL, CP64. For example, it is obvious that in this scheme, Phi plus and Phi D plus could be substituted by the reduced and the oxidized forms of a redox indicator dye. But These model experiments failed to be apt to picture the physiological circumstances for two main reasons. First, the cusp membrane of the Becquerel phenomenon, which is supposed to represent the delicate natural membranes, is a metallic or at least a half-metallic conductor belonging to a larger group of metal sulfides and metal oxides, which, like Kleinwise, are suitable to substitute the cusp membrane in Becquerel's experiment. But so far, there is no reason to believe that natural membranes composed of organic material behave like metal conductors or can substitute metallic electrodes. Second, it often has been neglected in interpreting the Becquerel phenomenon that in order to reduce the Cu ions to metallic Cu and to oxidize SS, energy has to be liberated. According to Freundlich and Zollner, this energy could be made available by a porous structure of the cusp membrane. It would mean that a circuit current is established in the membrane, one part of which, due to metallic electronic conduction of Qs, passes across its substance, the other part across the pores, which are accessible to the free ions Na and No. Therefore, returning to the studies of Friedenwald, Stieler, and Flexner, it appears that the manner of interpreting their observations cannot be agreed upon. However, instead, another way opens, which is suggested redox potentials and tissue activity. Applying the technique of redox indicators, it has been shown that significant redox potential differences, or the lack of them, become evident wherever one compares the respiratory activity inside and outside single cells, amoebae, ova of echinoderms, plant cells, and others, or at different parts of an active organ. The latter behavior is especially marked in studying the developmental onset of function. Thus, according to Flexner and Stieler, the fetal chorioidal plexus, in its presecretory phase, displays neither the aforementioned shift of diastoph cations and anions, nor any difference in the intensity of the indophenyl blue reaction between the stroma and the epithelium, which would indicate a different distribution of indophenol oxidases, cytochrome oxidase, as a sign of a gradient of respiratory activity. See the epithelium and is present there in a greater concentration. In addition, in recent studies, Friedenwald, it succeeded in separating the two major histological structures of the chorioidal plexus from each other, and showed that the cytochrome oxidase dehydrogenase fumarase system, CHAP 27, is located almost entirely in the EJ epithelial cells. Thus, these biochemical findings can be correlated with the results of the redox indicator method. Also, it has been shown by Flexner that using the kidney of fetal pigs of different ages at the presecretory stage, only allowable and a highly distributed content of cytochrome oxidase can be detected, whereas in the secretory stage, marked differences appear between the physiologically active portions, the proximal tubules, parts of Lenneau's loops, the distal tubules on the one hand, the glomeruli and collecting ducts on the other. All these gradations in reactivity disappear during asphyxia, just as function all living tissues requiring aerobic conditions, according to Friedenwald, at the flow of water normally ensuring the turgor of the eye bulb, also falls off during asphyxia, a phenomenon strengthening the assumption of the authors that the shift of water is due to electroosmosis, somehow connected with the oxidoinduction potential differences. But all this suggests quite another solution of the main problem of this section, in paying attention to the fact that the energy liberated by metabolic reactions is utilized in different objects for various kinds of work. For example, in the kidney for the secretion of certain solutes in one segment of the tubules, for the reabsorption in another. The conclusion will be drawn that the result of the energy output is derived by the SJ-specific construction pattern of the acting machinery. It seems clarifying that, referring to the previously described model experiments of Zollner, P.P. and Guy furthermore, referring to the present theory of the Becquerel phenomenon, which does not involve the assumption of an electron shift, the following picture may be offered. The Olszalek and Kruger-Inns have demonstrated that if a circular canal is filled with electrolyte solutions, as pictured in figure 69, that is, with different sectors filled by solutions containing cations and anions of suitable concentration and mobility, and in direct contact with each other, a circular electric current flows detectable by a magnet suspended within the ring formed by the canal. In other U6, cations circulate in one anions in the other direction, as long as the concentration differences between the solutions persist. On the basis of this experiment, Solner has proposed a fluid circle as in Fig 70. X is a colloidal membrane with narrow HX with wide pores, AI. Through the prevailing permeability of QK is the site of an electromotive force which drives the fluid through high, the liquid junction potential of which is practically zero P3S17. This may be comparable to the situation in a sieve membrane, which, due to the variety of pore diameters, pages 238 through 18, could be the site of local circular electric currents, pages 621 if now, a basic dyestuff is added to the solution inside the canal above high, stroma, the dications will become visible below in the epithelium, migrating with the electrosmotic current of water. If an acidic dyestuff is added below 6I, the dye anions will appear above in the stroma. Dye ions, according to their size, may or may not be allowed to pass at heat. As suggested previously, P622, the activity of such a system could possibly be maintained by a supplementary provision of suitable ions below the sieve membrane. In living tissues, such conditions would be secured by the production of metabolic ions in the epithelium, e.g. 11 and HCO3, arising from carbohydrate metabolism. This interpretation, then, differs from that of Stieler-Friedenwald and Flexrier in that, instead of viewing the oxidation-reduction systems as directly producing a current of electrons, which forces the water in the solutes across the blood aqueous and blood cerebrospinal barriers, it considers these systems to be the source of ions which arise from metabolism and which, due to the selective ion permeabilities of the systems, originate an electric current.